Hello students, myself Archana, your botany mentor, and the chapter we are going to cover today is biological classification, right? I'll say that again, that before starting the lecture, grab your NCRTs and ready for the chapter to be done, okay? Underline the important lines along with me and learn the lecture along with me. Be focused throughout the lecture and your lecture would be ready for NEET, okay? So without delaying, let's start the chapter. So what chapter is saying? It's saying about classification. That means dividing the various organisms into groups. So what we are going to cover today, we are going to cover the different groups. Like we are going to discuss what is Monera, what is Protista, what is fungi, and what are viruses. Okay, so this is the goal of today's lecture. And now, as we are talking about the classification of organism, that means we have divided the organism into various groups, right? And the 5 groups in which we have divided the organisms are kingdom Monera, kingdom Protista, kingdom fungi, kingdom Plantae, and kingdom Animalia. This you will read in the zoology. This I have already taught you in the last lecture. So now we are left with Monera, Protista, and fungi. So are you ready to read all these chapters or topics? Okay, now first let's discuss about 5 kingdom system classification, because this is the system of classification which is most accepted system of classification, and you all know about it, right? So 5 kingdom system of classification, beta, it was given by R.H. Whitaker. R.H. Whitaker. Now, on what basis he divide all the organism into 5 categories? That is also important. So the basis of his classification, I'm writing down the points of NCRT here. First, it was cell structure. Whether it is a prokaryotic cell or a eukaryotic cell. So first basis of classification was the cell structure. Then second, body organization. That means whether the organism is unicellular or it is multicellular. Then comes the mode of reproduction. As you know, beta, that the lower organisms, they prefer to reproduce asexually, and the higher organisms, they prefer to reproduce sexually. So mode of reproduction was also the basis of classification. And then nutrition. Obviously, some of the organisms are autotrophs and some of the organisms are heterotrophs. So that was also the basis of classification. These all are the points of NCRT. Okay, these all are the points of NCRT, but I have not written the most important point up till now. And the most important basis of classification taken by Whitaker was, see important, beta, see here, it is important, phylogenetic relationship. It was not discussed before R.H. Whitaker. That is why the Whitaker system of classification is the most advanced one and is the most accepted one. Why? Because he has talked about a very special concept, and that is phylogenetic relationship. That means ancestral history. Or you can say evolutionary history. That means if we trace the organism, in the process of evolution, all those organisms which have the common ancestor, they can be grouped in one group, right? Because they have common ancestor. So the kind of study which deals with the ancestor of the organisms, with the evolutionary process of the organism, that is the phylogeny. That branch of science is called as phylogeny. So this question might come, who was the first scientist who took phylogeny as the basis of classification? And he was R.H. Whittaker. So this is the most important one. This is the most important one which you will not forget. Okay, phylogeny. Okay, now we understood that the R.H. Whittaker, the most acceptable and the most advanced system of classification which we follow, has divided all the organisms into 5 groups, right? Now there is a comparative table beta given in the NCERT, and this is important. Let's discuss this. This table is pretty important. Okay, so here learn this table along with me, and how question can come from this table, we'll discuss that also. So characters first. First of all, this table is comparing the cell type. In the kingdom Monera, all the organisms are prokaryotic. That means the primitive organisms, those who do not have a proper nucleus and cell organelles. But rest all the 4 kingdoms are eukaryotic kingdoms. Okay, rest all the 4 kingdoms are eukaryotic. Only Monera is the prokaryotic kingdom. Then cell wall. Now, in the kingdom Monera, the members of kingdom Monera, they have cell wall, but their cell wall is non-cellulosic. That means it does not have cellulose in it. Rather, it is composed of polysaccharide and amino acid. Polysaccharide and amino acid, which sometimes is also called as peptidoglycan. This is also called as peptidoglycan, right? So remember this beta, that the kingdom Monera has cell wall, but that is non-cellulosic in nature. It is composed of peptidoglycan, which is composed of polysaccharide and amino acid. Remember this? Okay. Now the members of Protista, some of the members of Protista, they have a cell wall, and some of them, they do not have cell wall, like amoeba, paramecium, all the protozoans, they do not have cell wall. But diatoms, dinoflagellates, they do have cell wall, right? So if what is written over here, that in the members of Protista, the cell wall is present only in some members, and those who have the cell wall has cellulose along with some other compounds that we will be dealing in the later part. Like diatoms, they have silica along with cellulose. The dinoflagellates, they have this pectose along with cellulose, right? So in Protista, cell wall is not necessarily present in all the members. Then fungi, yes, cell wall is present, and again the cell wall is non-cellulosic. Again the cell wall is non-cellulosic. That means it is composed of chitin. Chitin is a compound which is non-cellulosic in nature. Then Plantae, obviously the member of Plantae has a cell wall, and that is cellulosic in nature. And obviously the members of Animalia, they do not have cell wall. So if the question comes that in how many kingdoms cell wall is present, simply in how many kingdoms cell wall is present, so you will say Monera, Protista, Plantae and fungi. Then in how many kingdoms non-cellulosic cell wall is present? In how many kingdoms non-cellulosic cell wall is present? In 2, that is Monera and fungi, right? And Plantae have cellulosic, and this Protista, in few members cell wall is present, and that is cellulosic along with some other deposition. And in Animalia, cell wall is absent. I hope these is clear to all of you. Okay, now let's go to the nuclear membrane. Obviously, when a cell has a proper nucleus, it will be having nuclear membrane. But the prokaryotic cell, the bacteria, they do not have a proper nucleus, so they will not have a nuclear membrane. So it is absent. Rest all the kingdoms are eukaryotic kingdoms. They will be having a nucleus, so they will be having a nuclear membrane. So rest all of them would be having nuclear membrane. Okay. Now let's go to body organization. Body organization. In the kingdom Monera, what is written over here? They have cellular body organization. That means their body is composed of a single cell. That means they are unicellular organisms. In the Protista also, the body organization is cellular. They are also unicellular organisms. Then see, for the first time, multicellularity has come in the kingdom fungi. For the first time, multicellularity has come in the members of fungi. That means cells, when they come together, they form an aggregation called a tissue. And all those organisms which have a tissue, they are multicellular organism. But here, because the tissue has come for the first time, so the tissue is not properly formed and they have loose tissue. They have loose tissue. So if the question come, multicellularity has first come in which kingdom? Your answer will be fungi. And the fungi has loose tissue. Then obviously the members of kingdom Plantae are all multicellular, and the members of kingdom Animalia, they all are multicellular. But remember one thing, beta, that when we talk about the multicellular organism, right, cell form tissue, tissue form tissue system form organ, and organ system form organism, right? So how many levels are there? Cell to tissue, tissue to tissue system to organ, organ to organ system, and organ system to organism. Clear? In plants, the highest degree of organization which can be present is organ. Plants have tissues and organs, but the animals, they have organ, but organ system as well. We have the full organ system in us, which make us the organism, right? So the highest level of organization in animals is organ system. Clear? Organ system. So animals have tissue, organ, as well as organ system. So what kind of questions can come from here? Listen carefully. In how many kingdoms cellular organization is present? Your answer will be 2, Monera and Protista. In how many kingdoms multicellularity is present? 3, Fungi, Plantae, Animalia, right? In how many kingdoms tissue is present? Tissue. Listen carefully. In how many kingdoms tissue is present? So the answer will be Fungi, Plantae, Animalia. Clear? In how many kingdoms tissue system is present? 2, Plantae, Animalia. In how many kingdoms organ is present? 2, Plantae, Animalia. In how many kingdoms organ system is present? Only 1, and that is Animalia. I hope you understood it well. Now let's talk about mode of nutrition. You know, beta, that generally the organisms are autotrophs and heterotrophs, right? Heterotrophs cannot make their own food. Autotrophs, they can make their own food. Autotrophs further can be of 2 types. They can be photosynthetic or they can be chemosynthetic. Listen carefully. Some organisms, they can make their own food with the help of light energy, and they are called as photosynthetic. But few organisms have the power to synthesize their own food, not with the help of light energy, but with the help of chemical energy, and they are chemosynthetic. In the whole nature, God has given this chemosynthesis only in one kind of organism, and they are bacteria. Only bacteria can synthesize their own food with the help of chemical energy. So let's see what kind of nutrition is seen in different kingdoms. See, bacteria, some of the bacteria are autotrophic also. And when they are autotrophic, they can be chemosynthetic and they can be photosynthetic. See, some of the bacteria are photosynthetic and some of the bacteria are chemosynthetic also. And most of the bacteria are heterotrophs. And what kind of heterotrophs? Most of them are saprophytes. That means they take their food from dead and decaying matter. Clear? Now, Protista. The members of Protista again can be autotrophic or can be heterotrophic. But When the members of Protista are autotrophic, they can be only photosynthetic because chemosynthesis can only be seen in bacteria. Okay? Now, fungi. All the fungi are heterotrophic. They are not autotrophic. They derive their food from dead and decaying matter, and they can be parasites also. They can never be autotrophs. And obviously plants, all the plants are autotrophs. And what is the term we use for them? Just look here. The term we use for them is holophytic. Holophytic. What is the meaning of holophytic? The higher organisms which can synthesize their own food with the help of light energy, they are called as holophytic. And this word holophytic are used for all the plants like algae, brio, pterido, gymno, angio. Okay, remember this beta. This holophytic word means they are multicellular complex organisms which can synthesize their own food with the help of light energy. So the members of the kingdom Plantae, they are holophytic. Now Animalia. Obviously, the animals are heterotrophs. They cannot synthesize their own food. So they are heterotrophic. And what is the word we use for them? Holozoic. They are holozoic. They ingest the solid food. So the members of kingdom Animalia, they are holozoic in nature, and very few of them are saprophytic also. Clear? I hope you understand now that in what way you have to understand this table, in what way you have to learn this table, and what kind of questions can come. Clear? Okay. Now, some questions again. How many kingdoms have autotrophic mode of nutrition? You have 2 seconds. Think. How many kingdoms have autotrophic mode of nutrition? Done? Answer is 3. Monera, they can also be autotrophs. Protista can also be autotrophs, and kingdom Plantae can also be autotrophs. Fungi and Animalia, they all are heterotrophs. Next question. How many kingdoms have photosynthetic mode of nutrition? Photosynthetic. Answer is again 3. Monera, Protista, and Plantae. How many kingdoms can have only chemosynthetic mode of nutrition? Answer is bacteria, only 1. How many have heterotrophic mode of nutrition? How many kingdoms have heterotrophic mode of nutrition? Tell. 4. Monera, Protista, Fungi, Animalia. I hope you understand this question now. So let's move forward. NCERT has also discussed about 2 kingdom system of classification, which was given by Linnaeus, right? This is the most previous system of classification. Initially we used to classify organisms based on the use of organisms. Okay, when we used to live in the forest, when suppose these particular organisms give us food, okay, divide them in the category of food or shelter. So very earliest system of classification were based on the use of organism. Then Aristotle came. He gave the first scientific basis of classification. He classified plants into herbs, shrubs, and into trees. And he classified animals into enema and anema. Those who were having the red blood cells, they were enema. And those who were not having the red blood cells, they were anema. So the first scientific basis of classification was given by Aristotle. And then came Linnaeus, who gave the 2 kingdom system of classification. And the most advanced system of classification we read is 5 kingdom system of classification, which is most accepted because it talks about phylogeny, that is ancestral history. Okay, so let's discuss 2 kingdom classification. I hope up till now you have learned things with me, right? Chalo. Okay, so 2 kingdom system means only 2 kingdoms are there, Plantae and Animalia. Now what is important here to remember, what are the organisms that are included in the kingdom Plantae? All the bacteria, all the kinds of bacteria, simple bacteria or cyanobacteria, that is blue-green algae, all of them were in Plantae. Diatoms, dinoflagellates, euglena, fungi, all the kinds of algae, red, brown, green, brio, pterido, gymno, angio, all these were present in Plantae according to Linnaeus. And who were in Animalia? amoeba, paramecium, all the invertebrates and all the vertebrates, right? Now what you can see here, what I can see here is that all the bacteria, beta, all the bacteria are unicellular. All the bacteria are unicellular and prokaryotes. All the cyanobacteria are unicellular and prokaryotes. Diatoms are unicellular and eukaryotes. Dinoflagellates are unicellular and eukaryotes. Euglena, again unicellular, eukaryote. Fungi can be unicellular or can be multicellular, and they are also eukaryotes. On the earth, only bacteria are prokaryotes. Rest all of them are eukaryotes. Algae can be unicellular or multicellular eukaryotes, and all of them are multicellular eukaryotes. Okay, amoeba, paramecium, they are unicellular eukaryotes, and rest all of them, all the vertebrates and invertebrates, are multicellular and eukaryotes, right? So what you can see here, that there was a mixture. Unicellular were placed with multicellular and prokaryotes were placed with eukaryotes. So this system was not very acceptable, and finally it has evolved into the 5 kingdom system of classification which we follow today. Now, very important line of NCERT I am trying to emphasize here. Just see beta, very important lines of NCERT. Read with me carefully. Amoeba, paramecium and Chlamydomonas chlorella. You know amoeba is a unicellular organism, paramecium unicellular, Chlamydomonas is unicellular green algae. Chlamydomonas is unicellular green algae, and chlorella is also unicellular green algae, right? You have just seen beta here that in 2 kingdom system of classification, algae, that means Chlamydomonas and chlorella, and amoeba and paramecium, they were separated, right? Linnaeus separated amoeba, paramecium, because amoeba, paramecium were in the kingdom Animalia and the Chlamydomonas and chlorella were in the kingdom Plantae. So 2 kingdom system has separated them, but they were united in the kingdom Protista. When we are talking about the 5 kingdom system of classification, then in Protista, all the 4 of them are present in the same kingdom, that is Protista. Now, when I will make you understand what is Protista, then you'll see that Chlamydomonas, chlorella, amoeba, paramecium, all 4 of them are present in Protista. So this line is very important. 2 kingdom system has separated amoeba, paramecium, Chlamydomonas, chlorella, but the Protista has united all 4 of them, because all 4 of them are unicellular organisms, and all the unicellular eukaryotic organisms are present in the kingdom Protista according to Whittaker. I hope you are clear with this line, right? Now, one more question students are often confused in, very confused. I have just taught you the plant kingdom in the previous lecture, and I have, I have taught all the kind of algae in the plant kingdom, right? I have taught all the kind of algae in the plant kingdom, whether they are unicellular or multicellular. But now I am saying that according to Whittaker, all the kinds of unicellular eukaryotic organisms are present in the kingdom Protista. So please listen carefully. Listen carefully. It doesn't matter where we study the organism, because the algologists, they study algae in plant kingdom, but according to Whittaker, some of the algae which are unicellular are present in Protista. So it doesn't matter where I have taught you the Chlamydomonas and chlorella. It is important that you remember that according to Whittaker, all the unicellular eukaryotic organisms, whatever they are, wherever you study them, wherever you read them. But according to Whittaker, in the Protista, all unicellular eukaryotic organisms are included. I have taught you Chlamydomonas and Chlorella in plant kingdom in algae. But if the question comes that according to Whittaker, Chlorella and Chlamydomonas are present in which kingdom, your answer will be Protista. I hope you are clear with this line. It doesn't matter where you read them, where I have taught you them. It is important that according to Whittaker, they are present in Protista. Clear? So Chlamydomonas, Chlorella, Amoeba, Paramecium, all four of them are unicellular eukaryotes. This question has come in the NEET, and the question was, according to Whittaker, Chlorella is present in which kingdom? According to Whittaker, Chlorella is present in which kingdom? And most of the students have answered plant kingdom, because in NCERT also the example of Chlorella is given in plant kingdom. Most of the students have answered plant kingdom, but no beta, the answer will be Protista, because the question is saying according to Whittaker. So according to Whittaker, all the unicellular eukaryotic organisms are present in Protista. Chlorella, Chlamydomonas, both are unicellular algae and they are eukaryotes, so obviously in Protista. So please read the question carefully. If it is saying according to Whittaker, then all four of them are present in where? Are present in Protista. And yes, one more question comes that Spirogyra or Eulothrix are present in which kingdom? Spirogyra and Eulothrix present in which kingdom? Can you answer? Can you answer? Just think about it. Spirogyra and Eulothrix, both of them are multicellular green algae. They are multicellular green algae, so they are not present in Protista, they are present in the kingdom Plantae. Remember this thing, beta. I hope you are getting my point. All the unicellular algae, we have studied all the kind of algae in plant kingdom, but according to Whittaker, all the unicellular algae will come in Protista and all the multicellular algae will come in kingdom Plantae. I hope you get this point. This is pretty important point, okay, from the NEET point of view. So now we have made our background, and now let's deal with the kingdoms. The first kingdom we have to deal with is kingdom Monera. We know that in kingdom Monera, only bacteria is there. Kingdom Monera is the kingdom of bacteria only. And on the earth, the prokaryotic organisms are only bacteria, right? So bacteria can never be multicellular. Bacteria can never be multicellular. So bacteria is a unicellular prokaryotic organism which belongs to the kingdom Monera. And when I say that bacteria is prokaryotic, what does I mean? What does that mean? If I say they are prokaryotic, that means they do not have proper nucleus, nuclear membrane. Nuclear membrane is absent. They do not have a proper nucleus. They do not have a proper nuclear membrane. This is the meaning of a prokaryotic cell. They do not have membrane-bound cell organelles. You know what our cell organelles are, right? Cell organelles. They have their own membrane. So membrane-bound cell organelles are absent. But bacteria has one cell organelle. Bacteria has one cell organelle in it, and that is non-membrane bound. So if the question comes that cell organelle is absent in bacteria, would that be right or wrong? If the question simply says cell organelle is absent in bacteria, would that be false or true? That would be false, because bacteria has a cell organelle, and that is not membrane bound. That is a non-membrane bound cell organelle, and that is ribosome. But yes, all the membrane bound cell organelles are absent in bacteria, like mitochondria, like chloroplast, peroxisome, vacuole. These are absent. Nucleus, they all are absent, right? So yes, they have a one cell organelle which is non-membrane bound, and that is ribosome. And the size of the ribosome is 70S. 80S ribosomes are found in case of eukaryotes. There are special type of proteins called as histone proteins. There are special kind of proteins called as histone proteins which are responsible for condensing the DNA and forming the chromosome. That histone proteins are absent in bacteria. Histone proteins are absent in bacteria. Then cyclosis. What is cyclosis? Cyclosis is the clockwise or anti-clockwise movement of cytoplasm in the cell, which helps in the, in the homogeneous distribution into the cell. This cyclosis is also absent in prokaryotes. Okay. So they do not have a proper nucleus, they do not have membrane bound cell organelle, they do not have histone proteins, and they do not have cyclosis in them, right? So this is pretty important. That is why we call them as prokaryotes. Now, bacteria, they are the most abundant microorganisms found on earth. They are the first ones to come on earth, right? And they are ubiquitous or cosmopolitan in nature. That means they are found everywhere, almost everywhere. They are found in hot deserts, they are found in snow, they are found in soil, they are found in deep sea, almost everywhere, right? So that is why they are cosmopolitan or ubiquitous. Then this line is very important, line of NCERT, that the structure of bacteria is very simple, but they are very complex in behavior because they are found almost everywhere and they can cause various type of diseases, and their metabolism is so different. The bacteria which is found in desert has a different metabolism. The bacteria which is found in hot sulfur springs has a different metabolism. So they are very complex in behavior, but they are very simple in structure, and they exhibit the most metabolic diversity. This question is very important. Out of all the kingdoms, metabolic diversity is maximum in which kingdom? And the answer comes bacteria. What is metabolic diversity? That the chemical reactions occurring in their cell, right? The metabolism is of diverse nature, and that changes according to their habitat. So these lines are pretty important. They are simple in behavior and they are complex in behavior. See, I've just deliberately called this as wrong because question also come like this. They are complex in structure, simple in behavior. False. Okay, and the most metabolic diversity is seen in Monera. So I hope these things are clear to you. They are lines of NCERT. Now, NCERT says that on the basis of shape, we divide bacteria into 4 categories. The first bacteria is cocci. The first bacteria is cocci, which is spherical in shape. Cocci is spherical in shape, and what is special about this bacteria that they are always non-motile. They are always non-motile. Non-motile means flagella will always be absent in them, and they are the most, least found. You can say they are the least found. The least found shape of bacteria is coccus. Okay, so these bacteria will always be non-motile. They will never have flagella. If the bacteria has a rod shape or a cylindrical shape, that bacteria is a bacillus bacteria, and this is the majority of bacteria have this kind of shape. This question directly comes. Majority of bacteria have bacillus shape, and they can be motile, they can be non-motile. Then spirillum, if they are spiral shaped, then spirillum. Then vibrio, if they are comma shaped. If they are comma shaped, they are vibrio, right? So these are the 4 different shapes of bacteria. Remember, the majority, the most common type of shape is bacillus. and the always non-methyl coccus. Okay, now let's understand what actually a prokaryotic cell, a bacterial cell, looks like. The detail of this you have already read in the chapter cell. That makes my work easy, right? So yeah, the detail of this is present in the chapter cell. We tackle it in the chapter cell, but let's have a basic idea of a bacterial cell. So you know that the outermost layer of bacteria is called as cell wall. Cell wall, which I have just told you, it is non-cellulosic, composed of peptidoglycan. Non-cellulosic, composed of peptidoglycan. Then beneath the cell wall, they have cell membrane. They have cell membrane, right? Then you can see here that I have invaginated the plasma membrane into the cytoplasm. When the plasma membrane invaginates into the cytoplasm, this invagination of plasma membrane is called as mesosome, and this invagination of plasma membrane is also called as chromatophore. Okay, now beta, if the invagination of plasma membrane has respiratory enzymes associated with it, if the respiratory enzymes are associated with the invagination of plasma membrane, then it is mesosome. And if the photosynthetic pigments are associated, if the photosynthetic pigments are associated with the invagination of plasma membrane, then this is chromatophore, because they do not have mitochondria and they do not have chloroplast. So in place of them, their plasma membrane goes into the cytoplasm and form a structure that is mesosome and chromatophore. This question is favorite of NEET, and it has come numerous times, that which part in bacteria is responsible for photosynthesis? Your answer is chromatophore. Chromatophore. Which part in bacteria is associated with the respiration? Your answer will be mesosome. And there are additional function of mesosome also. You must have read it in the chapter cell. That is binary fission. It also helps in binary fission in the cell wall formation. And it also helpful in DNA replication. It is also helpful in DNA replication. So these are the functions of mesosome. It is helpful in respiration, it is helpful in DNA replication, cell wall formation, and binary fission. Chromatophore, obviously, it is responsible for photosynthesis because photosynthetic pigments are associated with it. So this question directly comes. Learn with me: photosynthesis, chromatophore, respiration, mesosome. And obviously chromatophore is not present in all the bacteria. Obviously it is present only few bacteria which can photosynthesize. So it is present only in photosynthetic bacteria. Chromatophore will be present only in photosynthetic bacteria, right? Okay, the reserved food material. The food is stored in the form of various forms like cyanophycean granule, in the form of rubisco bodies, in the form of sulfur bodies, and they are called as inclusion bodies, right? The reserved food material in the bacteria is called as inclusion body. And this here you can see, this is the DNA. This is the DNA. And what is the nature of DNA here? This is double-stranded circular DNA. This is double-stranded circular DNA which is naked. It is not surrounded by any nuclear membrane because nucleus is absent in case of bacteria. So we call it as nucleoid. We call this double-stranded circular DNA as nucleoid, and it is not covered by any nuclear membrane. Okay, now here you can see some surface projections. These are called as pili. They may or may not be present in bacteria. Some of the bacteria have pili, some of the bacteria they do not have pili. And what is the function of pili? They help in reproduction. The structures you can see here are fimbriae. These are brush-like projections present on the surface of bacteria, and their function is adhesion. They are responsible for attachment of the bacteria to the surface, and they also can be present or cannot be present in the bacteria. It's not necessary that all the bacteria will have fimbriae. Okay, this is the flagella. You know what is the function of flagella. It provides motility to the cell, right? Then flagella can be present or can be absent in the bacteria. Okay, now one more important thing which is left, and that is the ribosome. Remember, it has only one cell organelle, that is ribosome, and the size of the ribosome is 70S. Okay, so this is the basic structure of a prokaryotic cell or a bacterial cell. See, such a simple cell. It does not have any complex structures. Just have a DNA at the center, which is naked, not surrounded by any membrane, double-stranded circular DNA. It has ribosome, which is a non-membrane bound cell organelle, and the size is 70S. Mesosome, chromatophore only in photosynthetic forms. Then reserved food material called as the inclusion bodies. Then pili, help in reproduction, may or may not be present. Then this is also, there is one structure also called as plasmid. This you must have read in your biotechnology chapter, that plasmid is an extra chromosomal DNA present in bacteria. Obviously, it is not present in all the bacteria, so it is also may or may not be present in the bacteria. So this is a simplified version of a bacterial cell or a prokaryotic cell. Some of the bacteria, they have additional covering around their cell wall, and that is Glycocalyx. What is Glycocalyx? Glycocalyx is also called as capsule or slime layer. The detail of this is present in the chapter cell. That Glycocalyx can be called as capsule or slime layer, is present in some bacteria, is present outside the cell wall, right? So the bacteria have an additional layer outside the cell wall, which is not necessarily present in all the bacteria, right? It is not necessarily present, may or may not be present in the bacteria. Clear? So I hope you understand what bacteria is now. Okay, now let's understand nutrition in bacteria. Yes, how bacteria gets their food. Nutrition in bacteria, most of the bacteria are heterotrophs. That means they cannot prepare their own food. And if they are heterotrophs, they can be parasites or they can be symbiotic. That means they can be associated with some organism and derive their food from them. But mostly they are saprophytes. That means they derive their food from dead and decaying matter. Then, yes, few of the bacteria, some of the bacteria are photosynthetic also. And some of the bacteria are chemosynthetic also, which prepare their own food with the help of chemical energy. And how that chemical energy get released? There are some inorganic compounds, not organic, inorganic. There are some inorganic compounds present in soil like ammonia and nitrate. They oxidize these inorganic compounds, release the energy, and use that energy in the synthesis of food, right? So these are the various kinds of modes of nutrition found in bacteria, but most commonly they are saprophytes. Okay, now let's discuss the photosynthetic bacteria, right? I have just told you, if the bacteria are photosynthetic, they have a special structure called as chromatophore. They have a special structure called as chromatophore, and chromatophore has photosynthetic pigments associated with it, and those photosynthetic pigment are bacterioviridin and bacteriopurpurin. That means, remember beta, that means photosynthetic bacteria, they do not have chlorophyll. The different kinds of chlorophyll A, B, C, D, E, these kind of chlorophylls are absent in bacteria. The photosynthetic pigments found in bacteria are two types: bacterioviridin, bacteriopurpurin associated with chromatophore. Bacterioviridin is green colored and bacteriopurpurin is purple colored. You must have heard that some bacteria are purple bacteria, some bacteria are green bacteria. All those bacteria which are purple and green, they are photosynthetic because they have photosynthetic pigments in them. Now, what is special about these photosynthetic bacteria? You know that in plants, when we talk about a photosynthetic reaction, we say 6CO2 plus 12H2O gives C6H12O6, right? So what is the function of water in this equation? The water breaks down and releases its electron and proton. And because of this, oxygen is released in photosynthesis. So in plants, water acts as electron and proton donor. And because of water, oxygen is released during photosynthesis. But in bacteria, water is not used as electron and proton donor. Why? Because they have hydrogen sulfide as electron and proton donor. During photosynthesis, when water breaks down, then only oxygen gets released. And this kind of photosynthesis is called as oxygenic photosynthesis. But in case of bacteria, because water is not used as electron and proton donor but hydrogen sulfide, so in bacteria, anoxygenic photosynthesis is seen. And what is anoxygenic photosynthesis? Where oxygen is not released during photosynthesis. And why in bacteria oxygen is not released during photosynthesis? Because water is not used over there. Instead, hydrogen sulfide is used. That is why sulfur gets released during photosynthesis, not water. Clear? So, what is special about photosynthesis in bacteria? That they do not have chlorophyll. They have bacterioviridin or bacteriopurpurin, and they do not have water as electron and proton donor, but they have hydrogen sulfide, and the kind of photosynthesis found in bacteria is anoxygenic. Okay? So now let's move on. See, if the electron and proton source is hydrogen sulfide in bacteria, right, during photosynthesis, then those bacteria are called as green sulfur or purple sulfur bacteria. This is important because this is given in the chapter NCERT. These words are used in the chapter photosynthesis in NCERT, right? So, who is a green sulfur bacteria? Why it is green? Because it has bacterioviridin in it. I have just told you bacterioviridin is green colored. So why it is green? Because it has bacterioviridin. Why it is sulfur? Because hydrogen sulfide is used as electron and proton donor. And the famous example is chlorobium. Why it is purple? Because it has bacteriopurpurin. And electron proton source is hydrogen sulfide. And the example is thiospirulum. Then, if the electron and proton source is not hydrogen sulfide but some organic compound which do not have sulfur in it, then the photosynthetic bacteria is purple non-sulfur or green non-sulfur. Green, bacterioviridin. Non-sulfur, electron proton source is not hydrogen sulfide but some organic compound which do not have sulfur. So non-sulfur. Okay. Example is chloronema. Purple non-sulfur, rhodospirulum, rhodopseudomonas. This name is important because it is given in NCERT in the chapter Mineral Nutrition. This is also a nitrogen fixer. Who is a nitrogen fixer? Who can fix atmospheric nitrogen and convert it into ammonia? Okay. So these are the photosynthetic bacteria. Photosynthetic bacteria can be green sulfur, purple sulfur, green non-sulfur, purple non-sulfur. Okay. And the examples are also given. So these all are the photosynthetic bacteria. Now comes the chemosynthetic bacteria, which can synthesize their own food, but not with the help of light energy, but with the help of chemical energy. And examples are hydrogen bacteria, hydrogenomonas, sulfur. Remember this thing, beta. This is very important. Sulfur bacteria. Now, some of the students will ask me, you've just told us that sulfur bacteria is photosynthetic bacteria. No, it was not just sulfur. It was green sulfur. There is difference when I just say sulfur bacteria and when I say green sulfur or purple sulfur. Green sulfur bacteria is photosynthetic. Purple sulfur bacteria is photosynthetic. But simply sulfur bacteria, simply sulfur is not photosynthetic. It is chemosynthetic. Because in sulfur bacteria, there is no photosynthetic pigment. And because it does not have photosynthetic pigment, it takes the help of chemical energy. Remember beta, simple sulfur bacteria, chemosynthetic. Green sulfur, purple sulfur, photosynthetic. Okay. And the examples are thiobacillus. This is important because the example is given in the chapter Mineral Nutrition. And this is denitrifying bacteria. The function of denitrifying bacteria is to convert the nitrates again into nitrogen, that is N2 form. Iron bacteria, ferrobacillus, is also a chemosynthetic bacteria. The most important ones are the nitrifying bacteria. Those bacteria which convert ammonia first into nitrite and then nitrite into nitrate, right? These bacteria are called as nitrifying bacteria and they are chemosynthetic. And the four most important examples are Nitrosomonas, Nitrococcus, Nitrocystis, Nitrobacter. Okay, so in this slide, whatever is written, whatever examples are written, they all are chemosynthetic bacteria. Okay, remember these examples. Let's move on further. Now what kind of questions come from this portion? There's a list of bacteria here and you have to find out how many of the bacteria are autotrophic. Autotrophic. That means how many of them can prepare their own food. That means you have to find out chemosynthetic as well as photosynthetic. Because the question is not asking choose only photosynthetic bacteria or choose only chemosynthetic bacteria. The question is asking choose autotrophic bacteria. That means both chemosynthetic and photosynthetic. So let's choose. Pseudomonas. Pseudomonas, no, it is heterotrophic. Pseudomonas is heterotrophic. Thiobacillus, yes, it is chemosynthetic. Rhizobium, no, it is heterotrophic symbiotic bacteria. Nitrosomonas, yes, it is chemosynthetic. Rhizobium again, sorry. Nostoc, Cyanobacteria, it is photosynthetic. So how many are autotrophic? 1, 2, and 3. 3 of them are autotrophic. Okay, so read the question carefully. The question is asking how many of them are chemosynthetic bacteria, or it is asking how many of them are photosynthetic bacteria, or it is asking how many of them are autotrophic. If the question is asking autotrophic, you have to count both photosynthetic and chemosynthetic. So this is the nutrition in bacteria. I hope it is clear to you. See beta, then we will move forward. I hope this much is clear to you. Let's move on to classification of bacteria now. NCERT has classified bacteria into two major categories. One is Archaebacteria and the other is Eubacteria. Eubacteria is also called as true bacteria. It is also called as true bacteria. Now, what are the major differences between Archaebacteria and Eubacteria that they has been characterized? One is the difference in their cell wall. The first difference lies in their cell wall. Archaebacteria, the cell wall of Archaebacteria is composed of pseudomurine. It is composed of pseudomurine, while the cell wall of bacteria or Eubacteria is composed of murine. Murine is muramic acid. I have just told you that in the cell wall of bacteria we have polysaccharides, and the polysaccharide is N-acetyl muramic acid, that is also called as murine. So in case of Eubacteria, murine is present, but in case of Archaebacteria, pseudomurine is present. So first difference is in their cell wall. Second difference is in their cell membrane. Archaebacteria has monolipid layer in the membrane, and the rest of the bacteria, the true bacteria, they have bilipid layer in their membranes. Generally, you have read this in the chapter Cell, that generally the structure of plasma membrane has two lipid layers. That is why it is bilipid layer. Every organism on the earth, the plasma membrane, the cell membrane has bilipid layer in it, but they are the exception. Archaebacteria are the exception. They have only single layer, single lipid layer, that is monolipid layer. Okay, so these are the two differences between Archaebacteria and Eubacteria. Now let's discuss Archaebacteria. Archaebacteria is also called as living fossil. Why it is called as living fossil? Because there are no changes in it till it has come on the earth, right? Archaebacteria can be easily found in the harsh conditions. Why they can be easily found in the harsh conditions? Because of the special structure of cell wall and cell membrane. The lipids, the lipid in the plasma membrane of Archaebacteria are highly branched lipids. They are highly branched lipids, and that is why they can easily tolerate harsh conditions. So this is the point of NCERT beta, which you don't have to forget, that what is the reason that Archaebacteria are easily found in harsh condition of high temperature, high acid and high salt concentration? Because they have special cell wall structure and they have special cell membrane in which the lipids present are highly branched. Okay, so Archaebacteria are sort of special bacteria easily found in harsh conditions. Now Archaebacteria are further classified into three categories. They can be Methanogens, they can be halophiles, and they can be thermoacidophiles. They can be thermoacidophiles. Okay. Now the name suggests itself, methanogen, the bacteria which generates methane. Okay, so they are, let's first discuss where they are found. Methanogens are found in marshy areas, or also called as the swampy areas. They are found in rice or paddy fields. They are found in rice or paddy fields. They are also found in symbiotic association in the rumen of cattle. They are found in the stomach of cattle, in symbiotic association in cattle, right? In stomach of cattle. The part of stomach of cattle is called as rumen. So they are found in rumen of cattle. Okay, so where methanogens are found? They are found in marshy areas. They are found in rice or paddy fields. And they are found in the rumen, the stomach of the cattle in symbiotic association. Where halophiles are found? They are found in the areas of high salt concentration. They are found in the areas of high salt concentration. Thermoacidophiles are found in the areas of high temperature. They can easily tolerate approximately 80 degrees of temperature very easily. And they are found in high acidic concentration. They are found in high acidic condition. And high acidic conditions means low pH. Right? So, these are different kinds of Archaebacteria which are found in harsh conditions. These are found in these conditions. This is found in high salt concentration, and this is found in the areas of high temperature and high acid concentration. Now, the Methanogens are obligate anaerobes. This point is pretty important. They are obligate anaerobes. Now, what is the meaning of obligate anaerobes? Obligate. Wherever the word obligate comes, that means they can be only anaerobes and nothing else. That means methanogens are found only and only in the areas where oxygen is absent. They are never found in the areas where oxygen is present. On the contrary, halophiles are facultative anaerobes. They are facultative anaerobes, and they also are facultative anaerobes. Okay? That means they are generally aerobic. Facultative anaerobes means they are generally aerobic. They are generally found in the areas of oxygen, but they can also survive in the absence of oxygen, right? So these are obligate anaerobes, and these two are facultative anaerobes. Methanogens are also helpful in biogas production. You know that biogas is a mixture of methane and other gases. Main component is methane. That is why they are methane producing bacteria. Okay? Is it clear? So this is the classification of Archaebacteria. Right? Archaebacteria has some eukaryotic characters also. Very interesting. Archaebacteria has some eukaryotic characters in it also. And what are they? Archaebacteria has histone proteins. See, I told you that histone proteins are absent in bacteria, but in Archaebacteria they are present, and they have introns in DNA. See, that means Archaebacteria is a sort of special type of bacteria. It can survive harsh conditions. It has histone proteins. It has introns in DNA. Introns are the part which cannot express themselves, but they are found in the DNA of eukaryotes, not in the DNA of prokaryotes. So, these are the eukaryotic characters found in Archaebacteria. Clear, beta? So, this is why Archaebacteria is a sort of special bacteria. So, what we have read till now about Archaebacteria, its cell wall is different, its cell membrane is different, and because of this, they can survive harsh conditions. They can be categorized into 3 categories, and these are they are also called as living fossil, and Archaebacteria also have some eukaryotic characters. Now let's move on to Cyanobacteria. Right, now let's move on to Eubacteria. Eubacteria or true bacteria. NCERT says that all the Eubacteria has a rigid cell wall. All the true bacteria has a rigid cell wall composed of peptidoglycan or murine, also called as murine. Right. Then they can be, the Eubacteria can be motile or they can be non-motile. That means they can have flagella or they cannot have flagella. And Eubacteria in NCERT beta, Eubacteria is of 2 types. Eubacteria, most of the bacteria are Eubacteria, but in the NCERT, in your syllabus, there is description of only 2 Eubacteria, that is Cyanobacteria and Mycoplasma. Cyanobacteria and Mycoplasma. So we'll discuss in detail what is Cyanobacteria and Mycoplasma. These are the 2 types of Eubacteria found. Yes, beta. So let's first move on with Cyanobacteria. Cyanobacteria, as you know, that is also called as blue-green algae, right? Now, important part is where blue-green algae is found. According to NCERT, blue-green algae is found in fresh water or marine water, or it can be found in terrestrial conditions, that is in moist soil. Okay, remember habitat, mainly it is aquatic, mainly freshwater or marine, but sometimes may found in moist soil also. This is habitat of Cyanobacteria. Now, Cyanobacteria, obviously, as it is a bacteria, it will be unicellular, no doubt. But sometimes Cyanobacteria can form a colony, and that colony can take a filamentous shape. So that Cyanobacteria will be called as filamentous Cyanobacteria, which will form a colony. Many unicellular Cyanobacteria comes and they form a colony, right, which can take a shape of a filament, and filamentous blue-green algae is nostoc and Anabaena. Another important feature of Cyanobacteria is the cell wall. The cell wall of Cyanobacteria, as it is a Eubacteria, it is rigid, composed of peptidoglycan. And one thing which is peculiar of Cyanobacteria, which is found only in Cyanobacteria, is the presence of mucilaginous or gelatinous sheath around the cell wall. Remember this. Presence of mucilaginous sheath around the cell wall is unique about Cyanobacteria. And this question has come in NEET. What is the nature of the cell wall of Cyanobacteria? And Cyanobacteria is always non-motile. That means flagella is completely absent in Cyanobacteria. Remember this point. Flagella is absent in Cyanobacteria. Okay? Then the Cyanobacteria or the blue-green algae is photosynthetic in nature. They can use light energy to form their own food. And some of them are nitrogen fixers also. Mostly or all of them, nearly all of them are photosynthetic, but some of them can be nitrogen fixers also. Those have the capacity to convert atmospheric nitrogen into ammonia. Okay? Then they have a certain type of structures in them called as gas vacuoles. Remember this, beta, that these are not true vacuole. A true vacuole is found only in a eukaryotic cell. But this is a pseudo vacuole. This is not a proper vacuole having its membrane. No, this is a pseudo vacuole, just a deposition of, just a compilation of gas. So it can provide buoyancy to the Cyanobacteria to come on the surface. Okay, so what we have read till now, Cyanobacteria, mostly aquatic, freshwater, marine, sometimes terrestrial. They are all unicellular, but maybe colonial also. Flagella completely absent, cell wall rigid, and may have, and yes, is surrounded by mucilaginous sheath. All of them, mostly all of them are photosynthetic, but some of them are nitrogen fixers. And gas vacuoles, they are pseudo vacuoles which provide the buoyancy. Okay, let's move on further. Yes, because Cyanobacteria is photosynthetic, then it must be having chromatophore. Remember, just told you chromatophore is responsible for photosynthesis. So in Cyanobacteria, the photosynthetic pigments associated with chromatophore are chlorophyll A. This is very important. This is very important. I have just told you that in bacteria, chlorophyll is not present. Bacterioviridin, bacteriopurpurin is present. But Cyanobacteria is the exceptional case. It is the only bacteria having chlorophyll A. Remember this. And the other type of photosynthetic pigment present in Cyanobacteria is phycobilin proteins. They are further of 2 types, C-phycocyanin and C-phycoerythrin. Phycocyanin is blue colored, Phycoerythrin is red colored. The amount of phycocyanin is much more. So blue. Chlorophyll A gives the green color. That is why it is called as blue-green. And like algae, it is found in water. That is why blue-green algae. Got the idea why they are called as blue-green algae? Phycocyanin and chlorophyll A. And let me tell you one thing. I have told you this in plant kingdom also, that phycobilin proteins are found only in cyanobacteria and second in red algae. Now, just told you that in bacteria, water does not act as electron and proton this donor. That is why in bacteria, oxygen does not come out during photosynthesis. But blue-green algae is an exceptional case where water acts as electron and proton donor and oxygen come out during photosynthesis. That means rest of the photosynthetic bacteria, they have anoxygenic photosynthesis, but cyanobacteria perform oxygenic photosynthesis. They are the first organism on the earth to add oxygen to the atmosphere of earth. Before them, the atmosphere of earth was reducing and they added oxygen. They first time added oxygen to the atmosphere of earth. We should be thankful to them. Okay. So, because water acts as electron and proton donor. So remember, what is important, what is exceptional about blue-green algae? That it has chlorophyll A and special proteins called as phycobilin proteins. Then water acts as electron proton donor, and that is why oxygen comes out during photosynthesis. Clear? Now let's move further. This is the diagram of nostoc given in NCERT. This is a colony of nostoc, right, which takes a shape of filament. So filamentous colony of nostoc. What you can see over here, you can see that, there is a mucilaginous sheath around the cell wall, which is a very unique point about cyanobacteria, right? All the cells of the colony, are called as the vegetative cells. The cells of the colony are called as the vegetative cells, which perform photosynthesis, which is also called as CO2 fixation, right? Photosynthesis is also called as fixation of CO2. But as you can see that in the colony, there is a special cell called as heterocyst. This cell is a specialized cell in the colony. This is a specialized cell, a modified vegetative cell which performs nitrogen fixation. Remember this. And what is nitrogen fixation? Conversion of N2 into ammonia. That means nostoc is a nitrogen fixer. These rest of the cells in the colony, it performs photosynthesis. But the specialized cells of the colony, that is heterocyst, they perform the function of nitrogen fixation. Clear? This is important. Now, some examples of cyanobacteria. The very famous examples are nostoc and Enamina. And what is special about them? That they can fix nitrogen in free living as well as in symbiotic condition. That means when they are present in the soil, they can fix the nitrogen, and when they are associated with some organism in symbiotic association, they can fix the nitrogen. So this is special. They are free living nitrogen fixers as well as symbiotic nitrogen fixers. Then Olocera, it's very important. It has come in NEET. Olocera and Tolipothrix, these are the two cyanobacteria which are found commonly in the rice or paddy fields, and they are free living nitrogen fixers. They are not found associated with rice. They are just present in the rice field and they can fix nitrogen in the free living condition. So they are free living nitrogen fixers found in rice or paddy fields. Important question from the point of NEET. Okay, then spirulina, and students often confuse it with spirogyra. Our eyes read spirulina, but mind gives the impression of spirogyra. No, spirogyra is multicellular green algae. This is unicellular blue-green algae. Okay. So spirulina is a blue-green algae which is used as single cell protein, SCP, because it has high content of protein. It is used by space travelers as a protein supplement. One more algae is used as a protein supplement. Can you tell me? I have taught you this in plant kingdom. Chlorella. Chlorella is a unicellular green algae. This is blue-green algae, and both of them are used as single cell protein. Now, Microcystis, not Macrocystis. Macrocystis is brown algae, okay? This is Microcystis. Microcystis and Oscillatoria, these are responsible for algal blooms. What are algal blooms? In the water body, sometimes in the polluted water body, right? Water body which has amount of sewage in it, that is nutritious for these algae, blue-green algae. They grow extensively in the water body and ultimately cover the whole water body and ultimately lead to the death of the aquatic lifestyle, the aquatic aquatic life like fishes and all. Okay, clear? So let's move further. Now let's go to another type of Eubacteria which is given in your syllabus, and that is Mycoplasma. I have listed all the important points which has come in NEET. See here, what are those important points? Mycoplasma is the bacteria which does not have cell wall. Very important point, very important point. Cell wall is absent. Flagella is completely absent. They are all non-motile. See what I have written here, DNA, double-stranded linear. The DNA of rest of the bacteria is double-stranded circular, but this is double-stranded linear like us. Penicillin is an antibiotic which kills bacteria, but Mycoplasma does not get killed by penicillin because penicillin is the antibiotic which attacks on cell wall and it does not have cell wall. So Mycoplasma is insensitive to penicillin. That means penicillin cannot do anything to Mycoplasma. But other antibiotics like chloramphenicol, erythromycin, streptomycin, they can harm Mycoplasma, but not penicillin. Yes, this is the smallest prokaryote. This is the smallest living organism on earth and smallest bacteria or smallest prokaryote also. They are facultative anaerobes. What is the meaning of facultative anaerobes? That means they are generally aerobic. They are generally found in the areas where oxygen is present. But if oxygen is not given to them, they can also survive in the absence of oxygen. That means they are facultative anaerobes. They can survive in the absence of oxygen also, right? They are also called as jokers of plant kingdom. Why? Because they do not have cell wall and they keep on changing their shape. They do not have a definite shape. And that is why they are called as pleomorphic. Pleo means variable. Morphic means morphology. They have variable shapes. They are pathogenic bacteria and they can cause disease both in animals and plants. And they all are heterotrophs, right? And moreover, they are all saprotrophs. Mostly they are saprotrophs. Okay? So these are very important points regarding Mycoplasma. What? Learned with me? Cell wall absent. Flagella absent. DNA double-stranded linear. They are different from rest of the bacteria in having double-stranded linear DNA. They are smallest prokaryotes. Okay? And they are pleomorphic organisms. Clear? So this is Mycoplasma. Now let's discuss some reproduction, how reproduction occurs in bacteria. Commonly, as you all know, bacteria prefers to reproduce by binary fission. But binary fission occurs in bacteria only under favorable conditions, right? So this is the NCERT diagram which is mentioning a dividing bacteria. This is a dividing bacterium. It is getting divided into two bacteria, right? So this is a diagram showing the binary fission. But under unfavorable conditions, what happens in bacteria? Endospore formation. Binary fission, favorable condition, but under unfavorable condition, endospores are formed, which are thick-walled structures and help the bacteria to pass on the unfavorable conditions. Yes, one more thing. In bacteria, true sexual reproduction is absent. What is sexual reproduction? First, formation of gametes, right? Second, fusion of gametes. And third, formation of zygote, right? Nothing of this sort happens in bacteria. In bacteria, gametes are not formed, fusion is absent, zygote formation is absent. That means true sexual reproduction is absent in them. NCERT says that they have sort of sexual reproduction. They reproduce by sort of sexual reproduction, which looks like sexual reproduction, but it is not sexual reproduction, right? What is the purpose of sexual reproduction? DNA mixing to create a new combination, right? Bacteria affords this new combination by primitive way of DNA transfer, right? They can transfer their DNA by very old method, by very primitive method, and because of that they manage to have some recombination. Because if they will keep on dividing, because if they will keep on reproducing by binary fission, then all of the bacteria will be the same. So during the course of evolution, they have found a way to transfer their DNA so that they can, create a new combination for variation. And this is not sexual reproduction. This is sort of sexual reproduction, which is a primitive method of DNA transfer, like conjugation, transformation, transduction. These are the three methods by which they can, cause some recombination or variation without actually going through sexual reproduction. So they reproduce by sort of sexual reproduction. Okay. So this is all about bacteria and kingdom Monera. I hope you understood it well. Read your NCERT line by line. Now we can, go ahead with the other topic, and that is Protista. So I hope you understand the bacteria well. So let's move on further, beta. Let's move on further to Protista. Let's continue with Protista now. Up till now, we've read that kingdom Monera has the unicellular prokaryotic organism. But the kingdom Protista, according to Whittaker, has all the unicellular eukaryotic organism. Unicellular eukaryotes. Now, this kingdom includes eukaryotes. What is the meaning of that? A eukaryotic cell has a proper nucleus. It has nuclear membrane. Remember, the Monerans, they did not have any nucleus and nuclear membrane, but they do have. They do have membrane-bound cell organelles. It was absent in the kingdom Monera. Histone proteins, which were absent in Monera, histone proteins are present here. True sexual reproduction was absent in Monera, but here the true sexual reproduction is present. And this kingdom shows great nutritional diversity because the members of Protista, they are heterotrophs as well as autotrophs. Now, this group, Protista, it does not have any specified boundary. Let me tell you one thing, that Protista is actually not a natural group. It is a made-up group. It is a made-up group to adjust all the unicellular eukaryotic organism. So it's not a natural group. It is a made-up group. In nature, someone is animal, someone is a plant. But Protista is a made-up group by the classification, taxonomists like the Whittaker. So it does not have any boundary because it includes all the kind of organisms in it. It has plant-like organisms, it has fungi-like organisms, and it has animal-like organisms. It has all kinds of organisms in it, and that is why it does not have a certified boundary, right? Then, yes, the members of Protista, they may or they may not have cell wall. I have just discussed that in the Whittaker table, right? And the locomotory structures present in Protista can be flagella, can be pseudopodia, and can be cilia. And some of the members are non-motiles also, which do not have any locomotory structures, right? And yes, one more thing, they have the cell organelle, the non-membrane bound cell organelle ribosome present in them, which is of size of 80s. In Monera, the size of ribosome was 70s. Clear? So these are the important characteristics of the kingdom Protista. Remember, this does not have any specified boundary because it contains all the kinds of organism, and it is not a natural group, it is a made up group and includes unicellular eukaryotes. Now we classify this Protista. The classification of Protista is done. It includes the plant-like organisms, which are called as Protistan algae. It includes fungi-like organisms called as Protistan fungi, and it includes animal-like organisms called as protozoans. So the plant-like protistans are diatoms, dinoflagellates, and euglenoids because they can prepare their own food. The fungi-like protistans are slime molds, and the animal-like protistans are called as protozoans, which are flagellated, ciliated, amoeboid, and sporozoans. So this is the whole kingdom Protista, which we are going to deal with. Let's start with the diatoms, the photosynthetic protist. NCERT says that the golden algae also called as desmids, and diatoms together they are called as chrysophytes. So this question may come directly. What are chrysophytes? So you can say they are desmids and the diatoms. Now, diatoms, the habitat of diatoms, NCERT says that they can be freshwater and they can be marine. That means they can be found in seawater also. Remember the habitat also. Now they are phytoplanktonic in nature. What are phytoplanktons? Phytoplanktons are microscopic photosynthetic organisms which float passively on the surface of water. Okay, microscopic photosynthetic organisms which act passively on the surface of water. They are phytoplanktons. Now, what is important from the NEET point of view regarding diatoms, and it was also a question in 2014 and 2015, the nature of cell wall of diatoms. Very important. The cell wall, see, the cell wall, this is the cell membrane, right? The cell wall of the diatoms is arranged as if it is a soap box having this lower lid and the upper lid. So the structure of cell wall has soap box like appearance. This question was there in NEET 2015. Which organism has soap box like cell wall arrangement? So diatoms. The upper lid is called as the epitheca and the lower lid is called as the hypotheca. So cell wall is arranged as epitheca and hypotheca, and this arrangement is called as soap box like arrangement. Second important thing regarding the cell wall of diatoms is the deposition of silica over the cell wall. They have heavy deposition of silica on their cell wall, and that is why their cell wall is called as siliceous. In the sand, the particles which illuminate, they are silica. That is why their cell wall is glassy and transparent, and the cell wall is indestructible, because when the organism will die, the cell wall, the silica will deposit. So the cell wall has, what are important points regarding the cell wall? It has soap box like appearance. It is siliceous in nature, glassy, transparent, and indestructible. Okay. Diatoms, remember, they do not have any flagella. They are non-motile organisms. They do not have flagella. Okay. Let's move on further. Then, yes, as we know that they are photosynthetic protist, so they must be having photosynthetic pigments, and they are chlorophyll A and chlorophyll C. Remember, we have, I have just told you that Cyanobacteria has chlorophyll A. It has started, the presence of chlorophyll A starts from Cyanobacteria, and after that it is present in all the photosynthetic organisms found on earth. Diatoms, dinoflagellates, euglenoids, algae, brio, pterido, gymno, angio. That is why chlorophyll A is the universal photosynthetic pigment. It is found everywhere now. So it has chlorophyll A and chlorophyll C. Where chlorophyll C first came? It came in diatoms. Okay, and along with the main photosynthetic pigment, they have accessory photosynthetic pigments, that is xanthophylls and carotenoids. Remember, chlorophyll A, chlorophyll C. Okay, because they are photosynthetic in organism, so NCERT says that diatoms are the chief producers of ocean, because they are marine in nature also, and they perform photosynthesis. Half of the photosynthesis found in ocean, so that is why they are chief producers of the ocean. Now, yes, I have just told you that when the diatoms, they float passively on the surface of water, but when they die, they go at the sea bottom and get deposited over there. So what is actually getting deposited at the sea bottom? That is silica, right? When they become dead, they accumulate at the sea bottom, and actually silica is getting accumulated because they have silica in their cell wall, and it has occurred for billions of years. So now they are accumulations of silica over the seabed, and that accumulation of silica over the seabed is called as diatomaceous earth or diatomite. Okay, you can see the diagram over here. This seems like a powder, a very porous powder, which is scratched from the sea bottom. Okay, and it is used in various purposes right now, right? It is a very hot product in the market. It is used, it is very porous in nature, and because it is very porous in nature, it is used for filtration purposes, filtration of oils, filtration of syrups. Okay, then it is used in metal polishing also, because it is a bit hard compound, so it is used for the polishing of metal. But sometimes it is written that it is used for diamond polishing. No, it will produce scratch on the diamond. It is not used for diamond polishing, it is used for metal polishing. Okay, yes, this has insulating properties also, so it is used as insulators in refrigerators. It is also used to make bricks, to make the room soundproof. It is used, nowadays it is used in many purposes. Some people, they intake the diatomite also to cure their digestive problems. Okay, so this is diatomaceous earth. So what is important about diatom? Flagella absent, found in seawater as well as the fresh water. Cell wall is important. Siliceous, soap box like appearance, glassy, transparent and indestructible. Right, they are called as chief producer of ocean. Photosynthetic pigment A, C. Diatomaceous earth, highly porous in nature and used in sedimentation purposes. I hope you have learned the topic with me. Now let's move on to dinoflagellates. As the name suggests, dino means two, flagellates means flagella. That means they are photosynthetic protists having two flagella. Flagella was absent in diatoms. Now, this is the diagram of NCERT, and you can see here that the body of the diatoms, it has the cell of the diatom, it has two kind of depression. One is the transverse depression and the other is the longitudinal. It has two kinds of cavities in its cell, two kinds of cavity. One is the longitudinal cavity, one is the transverse cavity, right? From that cavity, from that groove arises two flagella. The flagella which arise from the longitudinal groove is called as longitudinal flagellum, and the flagellum which arises from the transverse groove, it is called as transverse flagellum. And both the flagella are perpendicular to each other. Both the flagella, they lie perpendicular to each other. Okay, so this is important. And yes, remember the habitat of diatoms, freshwater and seawater. But dinoflagellates are mostly seawater organisms or marine. Okay, and they can be found, they can be of various colors. They can be yellow, green, red, depending upon the pigments they have. Clear? Okay, let's move further. Yes, cell wall. How the cell wall of dinoflagellates is important. The dinoflagellates have a cell wall made up of cellulose, and around that cellulose there is a rigid, tough covering of extra material called as pectose. and because of the rigid coat of pectose they have, that is why we call that dinoflagellates has thick, stiff cellulosic plates. This also was a question in NEET. Which organism has thick, stiff cellulosic plates? Dinoflagellates, because of the presence of a rigid theca composed of the pectose around their cell wall. Then they are also photosynthetic in nature, so they will also be having photosynthetic pigment similar to diatoms: chlorophyll A, chlorophyll C, xanthophylls and carotenoids. Okay, now let's move further. Yes, pretty important. The dinoflagellates, they divide rapidly by binary fission, and because of the rapid multiplication of dinoflagellates in the water body, the water sea, the tides, when the tides appear in the water, they appear as red because they have numerous amount of dinoflagellates in them. So this question is very important. Which organism cause red tides? As you can see in the diagram over here, these are the red tides. And why they appear as red? Because they have numerous amount of dinoflagellates in them, and those dinoflagellates are gymnodinium. You know, spell with me, gymnodinium and goniolax. Do not confuse it with Gracilaria and Gelidium. Students often confuse them because they all start with G. Gracilaria, Gelidium, red algae, Gymnodinium, goniolax, they are dinoflagellates. Numerous time in many PMTs this question has come. Gymnodinium, goniolax are the dinoflagellates which cause red tides. Okay. Now some of the dinoflagellates, they are bioluminescent also. What is the meaning of bioluminescent? They have their own light. They can produce their own light. And the examples of bioluminescent dinoflagellates are Noctiluca, Pyrodinium, Pyrocystis. These are the three dinoflagellates which are bioluminescent. As you can see here in the diagram, they are so beautiful. They produce their own light and they illuminate the whole sea. The tourist, these kind of, marine bodies are the tourist attractions where the people go specifically to see those diatoms which produce light and they illuminate during night. So here you can see the diagram of the sea where the bioluminescent dinoflagellates are present, illuminating the whole sea. Okay. Yeah. Some dinoflagellates are poisonous also. Like one of the species of goniolax, that is Goniolax catinella. This is a poisonous dinoflagellate. It produces a toxic substance into the water body, which is intaken by the fishes. And those fishes, when they are, eaten up by the humans, in humans it can cause paralysis, which is called as paralytic shellfish poisoning. Because generally the shellfishes present in the aquatic body, in the seawater, they ingest these dinoflagellates and they die. And when we eat these shellfishes, we can also get paralysis. So these dinoflagellates are poisonous also, right? So what we have read, let's learn in a single line. Dinoflagellates, two flagella. One is longitudinal, one is transverse, one is longitudinal, perpendicular to each other. They are mostly marine. Photosynthetic pigment, chlorophyll A, chlorophyll C. Dinoflagellates, they can be bioluminescent, that they can produce their own light. They can cause red tides. That is done by goniolax and gymnodinium, and they can be poisonous also. Clear? Let's move on to the another members, that is euglenoids. Euglenoids are found in stagnant fresh water, stagnant, which is not moving, fresh water. See, compare the habitat of all of them. Diatoms, fresh water as well as seawater. Dinoflagellates, mostly seawater. But euglenoids, stagnant fresh water. Okay. Now, what is special about euglenoids which make the question important? Cell wall was present in diatoms. Cell wall was present in dinoflagellates. But in euglenoids, cell wall is absent. This is important. In euglenoids, cell wall is absent. And around the plasma membrane, the cell wall is not there. So they have a flexible proteinaceous covering around their cell wall called as pellicle. They do not have cell wall, but they have pellicle around their cell wall, that is flexible proteinaceous covering. Then sexual reproduction is completely absent in euglenoids, right? Sexual reproduction was present both in diatoms and dinoflagellates, but in euglenoids, they completely reproduce by binary fission. Okay? Euglenoids also have two flagella. Diatoms, flagella absent. Dinoflagellates, two flagella. And euglenoids again, two flagella. One is short, one is long. You can see here in the diagram, one flagella. This is the diagram of NCERT. One flagella is short and the other flagellum is long. Okay? Yes. Now this is the diagram of NCERT depicting euglenoids. See beta, euglenoids have two flagella, which we have just discussed. Then they have a special point called as eye spot or stigma. They have eye spot or stigma. What is the function of eye spot or stigma in euglenoids? It is photosensitive in nature. It can sense the light. Okay. They also have a contractile vacuole in them, which helps in osmoregulation. They have pellicle, that is proteinaceous covering around their membrane. Okay. So this is euglenoid. Now what is important regarding euglenoid? That the photosynthetic pigment, they are also photosynthetic in nature. Chlorophyll A is universal. You don't have to remember or cram this thing, because chlorophyll is present everywhere after cyanobacteria. So chlorophyll A is there, but it has chlorophyll B. Diatoms, chlorophyll A, chlorophyll C. Dinoflagellates, chlorophyll A, chlorophyll C. But euglenoids, chlorophyll A and chlorophyll B. And what I have written behind this is very important. Having chlorophyll B is higher plant-like character. Remember this, having chlorophyll B is higher plant-like character. So this question often comes, that which member of Protista exhibits show higher plant-like character? Your answer will be euglenoids. And what is that higher plant-like character? Presence of chlorophyll B. Okay. Now, euglenoids have mixotrophic nutrition. What is the meaning of mixotrophic nutrition? That means if sunlight will be available to them, they will be photosynthetic. But if sunlight is not present, then they will not die. They will turn into heterotrophic organisms. And when they turn into heterotrophs, they can act as predators, those making prey of the small organisms, or they can act as saprobes, right? So this kind of nutrition is mixotrophic. In the presence of sunlight, photosynthetic. In the absence of sunlight, they become heterotrophs. Clear? So this is about euglenoids. Now let's move on to the another members of Protista, that is, so plant-like protistans are finished. Now next thing, that is fungi-like protistans, and they are called as slime molds. Slime molds are found on land. They are terrestrial. Up till now, diatoms, dinoflagellates, euglenoid, they all were aquatic, but they are terrestrial like fungi. They are found on land, and they all are saprophytic. Slime molds are heterotrophic or saprophytic, that they obtain their food from dead and decaying material. Flagella is absent from their life cycle. They do not have flagella. And one more important thing which is absent in them is cell wall. So two important points: flagella absent, cell wall absent, right? Cell wall was also absent in euglenoids. Now, this is important. When these slime molds, they have favorable conditions, that means they have proper food, moisture, everything. When they have favorable condition, there is a particular stage in their life cycle which comes, and that particular stage is called as plasmodium, which you can see over here. And please do not get confused this plasmodium with the plasmodium, which is a protozoan. This is not protozoan. This plasmodium is a stage. It's a stage of life cycle of slime molds. It's an aggregation. Aggregation means formed by aggregation of many slime molds. Many slime molds, they come and aggregate and they form an aggregation which is called as plasmodium. And this aggregation, you have to remember this, is formed under favorable conditions. This is given in NCERT. Plasmodium in the life cycle of slime molds forms under favorable condition. And what kind of structure is this? It is an aggregated structure which do not have cell wall. It is having naked protoplast. Naked protoplast, no cell wall. And this plasmodium, it glides over the dead and decaying matter. It glides over the dead and decaying matter and absorbs food from the dead and decaying matter. Okay? So this is understood? Now, what happens during unfavorable conditions? During unfavorable conditions, this plasmodium, this plasmodium undergoes a process called as fructification. What is fructification? Fructification means formation of fruiting bodies on the plasmodium. Formation of fruiting bodies on the plasmodium is called as fructification. And inside these fruiting bodies, these are the fruiting bodies formed over plasmodium when under unfavorable conditions, and they bear spores at their tips. Line of NCERT. The fruiting bodies, they bear spores at their tips. Okay? Now, the spores are important. The spores of slime molds are important because they possess cell wall. Remember this. Cell wall is absent in slime molds, but only one structure in their life cycle has the cell wall, and that is spore. So they possess true walls. The spores are highly resistant. That means they can survive the most bad or adverse condition. They are very resistant and they can survive up to hundreds of years. They can survive for several years, right? So this makes the spores of slime molds very special. And the spores are dispersed by air currents. The spores, it get dispersed by air currents when they get when they come on soil, they germinate to give rise to new slime molds. Okay, because they are terrestrial, because they are found on soil, so obviously they will not be dispersed with the help of water, they will be dispersed with the help of air currents. Now, what is important regarding slime molds? Learn with me. Slime molds, fungi-like, saprotrophic. Okay, then in slime molds, flagella absent, cell wall absent. In favorable conditions, plasmodium is formed. Under unfavorable conditions, fruiting bodies are formed on the plasmodium. These fruiting bodies, inside these fruiting bodies, spores are formed and they bear spores at their tips. These spores are special because they have cell wall, which is generally absent. They are very resistant. They can survive for many years and they are dispersed with the help of air currents. Okay, so this is slime molds. Now let's move on to the last category of the protistans, and that is protozoans. And protozoans are the animal-like protists. They are said to be as the primitive relative of animals. They are primitive relative of animals. That means animals have evolved from them. And they all are heterotrophs. They can behave as predators and they can behave as parasites. Now we divide protozoans into 4 categories, that is ciliated protozoans, flagellated, amoeboid, sporozoans. Let's first discuss the ciliated protozoans. The ciliated protozoans, they are aquatic in nature. They are found in water bodies, right? Some of the forms of ciliated protozoans, they are free living. Free living, that means they can live on their own. And some of the ciliated protozoans are parasites also. So both kind of forms are seen in ciliated protozoans, free living forms as well as the parasitic forms. The cilia present over the body of the ciliated protozoans, it helps in movement also by displacing the medium, and it helps in ingestion of food. It takes the food towards the oral cavity and help in ingestion of food also. Okay. So this is the diagram of paramecium, which is a ciliated protozoan, and it has thousands of cilia over its surface, which help in locomotion as well as in ingestion of food. You can see here that this is the oral cavity. This is the oral cavity of the paramecium from which it intakes the food, and behind the oral cavity it has the gullet. Behind the oral cavity it has the gullet in which the food gets stored, and then it goes to cytopharynx where it gets digested. Okay, then it has two kinds of nucleus. It has two types of nucleus, macronucleus and micronucleus. It was the question of NEET. The macronucleus, it regulates the metabolic activities in the paramecium, the metabolism, the chemical reactions occurring, but the reproductive process is controlled by the micronucleus. Okay, micronucleus, reproductive process. Okay, and it also has contractile vacuole, which is there for the osmoregulation. Okay, so what is important regarding ciliated protozoans? They are aquatic, they can be free living, they can be parasites, they have cilia, and they have oral cavity behind which gullet is present. Okay, they have two kinds of nucleus, macro, micro, macro metabolism, micro reproduction. Clear? Now let's come on to the next category, and that is amoeboid protozoans. The amoeboid protozoans, they can be freshwater, they can be seawater, and they can be moist soil. They can be found in fresh water, they can be found in marine conditions, and they can be found in the terrestrial conditions also, that is moist soil. Amoeboids have a special structure in them called as pseudopodia, which is also called as false feet. And this pseudopodia helps the amoeboid protozoans in locomotion and also in capturing their prey and engulfing the food into the cell. Now, these amoeboid protozoans, the marine forms, remember, only the seawater forms, only some seawater protozoans have a special structure around their cell, that is silica shell. And this is understood. In all the protozoans, cell wall will be absent, right? Because they are animal-like. That means cell wall will be present only in the two members of Protista, that is diatoms and dinoflagellates. Rest, everywhere cell wall is absent, and it was present in the spore of slime molds. Otherwise, cell wall is absent. Chalo. So this is important. Not the freshwater amoeboid protozoans, but the marine amoeboid protozoans have a special structure around their cell membrane, that is silica shell. Okay, they also can be free living, they can be parasites. Free living member is amoeba and parasitic member is enta-amoeba. Okay, so these are amoeboid protozoans. Now, flagellated protozoans. The flagellated protozoans again can be free living and can be parasites. The locomotory organ is flagella. Then the example of flagellated protozoan, the most common example of flagellated protozoan is trypanosoma, which causes the disease sleeping sickness. And the last one is sporozoan. What you have to remember here, that all sporozoans are endoparasites. What we have read up till now, that diatoms, dinoflagellates, sorry, I've gone too, behind. Yeah, what I have just discussed, that the amoeboid protozoans, the ciliated protozoans, and the flagellated protozoans, all of them can be free living or they can be parasites. But these can only be parasites, or that too not ectoparasites, not the surface parasites. All of them are endoparasites. Okay, and the spores formed in their life cycles are infectious in nature. Clear? And the most infamous or notorious member of sporozoan is Plasmodium, which causes the disease malaria. And this is not the Plasmodium we discussed in the slime molds. There, the Plasmodium was the name of a stage of life cycle, and this is the name of the organism. Clear? So I hope you are clear with Protista now. Clear? Not a natural group, do not have any set boundaries, have unicellular eukaryotic members. Cell wall is present only in diatoms and dinoflagellates. or in the spores of slime molds. Otherwise, cell wall is absent in all the members. Then plant-like are diatoms, dinoflagellates, and euglenoids. Then fungi-like are slime molds. And then animal-like are the ciliated, flagellated, amoeboid protozoans and sporozoans. Okay, so this is about the Protista. Now let's move on to the fungi. I hope you are clear up till here. Now we'll discuss the fungi. Now fungi. As you all know, beta, fungi, all the fungi are heterotrophs, right? And when I say that they are heterotrophs, that means they are mostly saprotrophs or saprophytic. That means they obtain their food from dead and decaying matter. And they can be, some of them can be parasites also, and some of them can have symbiotic association also. Some of them can have symbiotic association also. And what is that symbiotic association? That is mycorrhiza and lichen. These are the symbiotic association shown by fungi, mycorrhiza and lichen. Mycorrhiza is the association of fungi with roots of higher plants, where it is helpful in the absorption of water and minerals from the soil, right? Lichen is the association with algae, where algae does the function of photosynthesis, and rest of the function is performed by the fungi. Okay, so let's discuss about their habitat. Fungi is terrestrial. That means fungi is found on soil. It is found on soil. And what are the conditions that are, helpful? Moist conditions. Most of the fungal infections, they occur in rainy season. That means they like moisture, humidity, moist conditions, and temperature approximately 30 to 35 degrees centigrade. That is warm temperature, right? So the best season is July, August for their growth. But some of the fungi, they are also found in, in water also. Very few of the fungi are found in water also. That means very few are aquatic also. They are found in their spores are found in air. Some of the fungi, they are found on the horns or nails. They can grow on the horns and nails also, and they are called as keratinophilous fungi. Keratinophilous. Or they can grow on cow dung also. That is called as coprophilous fungi. Coprophilous fungi. Okay, so fungi, they are, they show great diversity in their morphology. How do they look? This is the line of NCERT. How do they look? They show great diversity in morphology. But there are 3 lines. Remember these 3 lines. Listen carefully. If the question says which kingdom show most metabolic diversity, answer will be Monera. Most nutritional diversity, again Monera, because it can show chemosynthesis, photosynthesis, they can be heterotrophic. After Monera, nutritional diversity is maximum in Protista, and morphological diversity is seen in the members of fungi. They are present on soil, right? Some of them can be coprophilous, keratinophilous. They are also ubiquitous or cosmopolitan like bacteria. They are also found almost everywhere. Clear? So yes, now let's talk about the body organization. Let's talk about the body organization of fungi. The fungi, the members of fungi, They can be unicellular and they can be multicellular. Unicellular, name of the unicellular fungi, learn with me. The unicellular fungi are yeast and Syncytium. Yeast and Syncytium. These are the names of unicellular fungi, and most of the fungi are multicellular. And if they are multicellular, what is the meaning of multicellularity? Fungi are the first organism to be multicellular, right? This Protista and Monera was unicellular. So they have loose tissue in them. They have loose tissue in them. And what is their body composed of? Their body is composed of many thread-like structures. Many thread-like structure. And each thread is called as hypha. And many hyphae together, the interwoven network. These hyphae, they get interwoven together. Imagine one single thread is hypha, and when many threads come together and form a network, that is called as mycelium. So a multicellular fungi has mycelium, which is an interwoven network of hyphae. So interwoven network of hyphae is called as mycelium. Right? So unicellular fungi have only one celled body, but the multicellular fungi, it has loose tissue, just composed of many threads, hyphae, and they interwoven together to form mycelium. Now, what is important? That this mycelium I'm talking about can be further of two types. Mycelium, if I take single thread of the mycelium, whatever is the nature of the single threads and the threads of the mycelium, the same nature would be of the mycelium, right? If I take one thread and see, I'm observing this thread, and in this thread, inside the thread, there are septas or cross wall dividing this thread into compartments. Then this kind of mycelium would be aseptate, this kind of mycelium would be septate mycelium. This kind of mycelium is septate mycelium. Okay, remember beta, septate mycelium. The mycelium would be having septa or cross walls, right? But if it does not have any cross walls or septa, then this kind of mycelium is called as aseptate mycelium. Aseptate mycelium. And you must be having the idea of this, that we will classify fungi into four classes: Phycomycetes, Basidiomycetes, Ascomycetes, and Deuteromycetes. So aseptate mycelium is found in the member of Phycomycetes. Rest all the three, that is Basidiomycetes, Ascomycetes, and Deuteromycetes, all of them has septate mycelium. Will you remember this? Aseptate mycelium, Phycomycetes, and septate mycelium, Deutero, Asco, Basidio. Clear? Now let's take this mycelium, the septate mycelium. Now let's discuss the septate mycelium. This septate mycelium further can be of two types. It can be monokaryotic or it can be dikaryotic. What is the meaning of karyon? What is the meaning of karyon? Karyon is nucleus. If this septate mycelium, each compartment of this septate mycelium have only single nucleus, then obviously this kind of mycelium is called as monokaryotic mycelium. each compartment has a single nucleus. But if each compartment has two nuclei in it, two nuclei, then obviously each compartment has two nuclei. So di means two, karyotic means nucleus. So this kind of mycelium is the dikaryotic mycelium. Clear? Now what a question I'm just discussing with you. What is a dikaryon? It has, it was in NEET. What is a dikaryon? How will you define a dikaryon? So obviously, a dikaryon is a structure having two nucleus together, having two nuclei together. It's a dikaryon, right? So a dikaryon is a structure having two nuclei together, and whether those two nuclei are similar or dissimilar to each other. Obviously, these two nuclei are unfused. They are not fused with each other. They are unfused and genetically dissimilar. Remember this thing, beta. A dikaryon is a structure where the two nuclei present, they are unfused, they are not fused, and they are genetically dissimilar. They are not similar to each other genetically. That means I will not write this structure as a 2n. Rather, I will write it as n plus n. I hope you understand this. I will not write it as 2n because they're unfused. I will write it as n plus n. So I hope you're clear with it. You can imagine fungi now. Fungi, unicellular, only two examples, East Syncatrium. Multicellular, composed of mycelium. That mycelium can be septate or aseptate. The septate mycelium further can be monokaryotic or dikaryotic, and you understand now that what is a dikaryon. Okay. Now, some other characters of fungi are their cell wall. What is the composition of cell wall of fungi? The cell wall of fungi is non-cellulosic, composed of chitin. And it is non-cellulosic in nature. Clear? So the members of fungi, the kingdom fungi, they have cell wall, and that is composed of chitin. Then reserved food material. In what form food gets stored in fungi? What is the reserved food material in fungi? In fungi, the food generally gets stored in the form of glycogen. Right? This is the animal-like character in fungi. Presence of cell wall is the plant-like character in fungi, and reserved food material glycogen is the animal-like character in fungi. Okay? So I hope all these things are clear. Now, the important part, which is favorite of NEET, is reproduction. Reproduction in fungi. This is favorite of NEET. Reproduction in fungi. Okay. First, let's discuss the vegetative reproduction. Vegetative reproduction in fungi, that occurs with the help of first binary fission, right? And you know that binary fission will be seen only in the unicellular forms. That means yeast or Syncytium. Second method of vegetative reproduction is fragmentation. And fragmentation is seen in fungi like Rhizopus. Then they can also show budding, and budding is seen in case of yeast, right? So these all are the methods of vegetative reproduction. Now let's see method of asexual reproduction. Right? How vegetative reproduction is different from asexual reproduction? Both of them are asexual reproduction, but in asexual reproduction there is formation of some specialized structure. So they reproduce with the help of some specialized structures. And those specialized structures are called as spores, asexual spores. Asexual spores. And this is the favorite topic of NEET. Please beta, pay attention towards this, because this is favorite point of NEET, asexual spores. Okay. Now let me tell you beta, that the body of fungi, the main body of fungi is haploid, because the life cycle of fungi is haplontic. So the main body of fungi is haploid, and on that haploid body, on that haploid mycelium, the asexual spores are formed by mitosis. So obviously, all the asexual, there are two kinds of spores found in the life cycle of fungi, sexual spores and asexual spores. Sexual spores are formed by meiosis, but the asexual spores are formed by mitosis. Remember this thing. So these are asexual spores here, right? So what is important regarding all the asexual spores? That they all are mitospores. Mitospores. What is the meaning of mitospores? That they all are product of mitosis. Clear? And the three main asexual spores found in the life cycle of fungi, three main asexual spores are zoospores, sporangiospores, and conidia. Learn with me. Zoospore, sporangiospore, and conidia. These are the three asexual spores. All of them are mitospores and help in asexual reproduction. Let's discuss them, because this is important with point of view of NEET. Zoospores. Obviously they are haploid, right? So zoospores are motile spores. Remember this. They are motile spores. That means they have flagella in them. Flagella is present in them. Remember this thing. Okay. Then zoospores are endogenous in nature. This is again important. Endogenous. That means they are not formed over the surface. They are formed inside some structure. So they are endogenous in origin. They are formed inside the zoosporangium. Okay. Inside the zoosporangium. So that is why they are endogenous in nature. Zoospores are dispersed by water currents. They are dispersed with the help of water currents. Now you can understand that these kind of spores will be found only in aquatic fungi. They are found only in aquatic fungi. Remember this beta, this is very important. They are found only in aquatic fungi. And the aquatic fungi are found only in the class Phycomycetes. Esco, Basidio, Deutero, they all have only terrestrial members. So aquatic fungi is, that is few members of Phycomycetes. Few members of Phycomycetes, right? This is important beta. Few members of Phycomycetes only. That means zoospores are not very common in fungi. They are found only in few fungi. That too, the fungi found in water. That means only few members of the class Phycomycetes. Remember, I have told you in algae that zoospore is the most common asexual spore of algae. That means, what does that mean? That means zoospore is rarely seen in fungi. And zoospore are also called as, the another name of zoospore is called as planospore. Why it is called as planospore? Because plano means motile, because it is motile, right? The second important spore, asexual spore, is sporangiospore. It is also called as aplanospore. Why aplanospore? Because it is non-motile. That means flagella absent. Flagella absent. They are non-motile spores. Okay, they are thin-walled. And they are endogenous in origin. That means they are also formed inside the sporangium. And where they are found, example, they are found in terrestrial members of Phycomycetes. The members of Phycomycetes which live on soil, they are found in terrestrial members of Phycomycetes, and they are rhizopus and mucor. I think this was a question of the year 2014 or 15. It was a question. There were 4 statements and incorrect statement was to be chosen. And one of the statement was mucor has biflagellated zoospore. So that statement is false or correct? Mucor has biflagellated zoospores. Obviously false. Zoospores are found only in aquatic members of Phycomycetes, but mucor is the terrestrial member of Phycomycetes. Clear? Now, the third important spore is conidia, or also called as conidiospore. Okay, conidia or conidiospore. Now, this conidia, obviously this is also non-motile, so flagella is absent in it. Then it is thick-walled. It is a thick-walled spore, and see what I will write over here, very important. This is exogenous. This is exogenous. That means it is not formed inside, it is formed at the surface. It is formed on the surface. So this is exogenous in nature. Okay. And it is found in the members of Ascomycetes and Deuteromycetes. And this is the most common asexual spore of fungi. Direct question comes. This is the most common asexual spore of fungi. Which is the most common asexual spore of algae? Zoospore. And the most common asexual spore of fungi? Conidia. So, in Phycomycetes, in the members of Phycomycetes, you can see zoospore and sporangiospore. Zoospore in the aquatic members and sporangiospore in the terrestrial members. In Ascomycetes and Deuteromycetes, you can see conidia, which is exogenous. Both of them are endogenous. Both of them are non-motile. This is motile. Clear? Okay. So, now you will say, ma'am, where is Basidiomycetes? You haven't mentioned Basidiomycetes. Beta, asexual spores are absent in Basidiomycetes. Okay? This is the line of NCERT. Asexual spores are generally absent in Basidiomycetes. Clear? So this part is very important regarding which is endogenous, which is exogenous, which is motile, which is non-motile, right? So these are very important. This is the favorite of NEET. Now let's discuss the sexual reproduction in fungi. How sexual reproduction occurs in fungi? According to NCERT, the first step in the sexual reproduction is the plasmogamy. Plasmogamy means fusion of cytoplasm. During sexual reproduction, there is fusion of two gametes, right? Or fusion of two structures. During that fusion, there will be first fusion of cytoplasm. Gamy means fusion, plasmo means cytoplasm. The second step will be karyogamy. Karyo means nucleus, gamy means fusion, that is fusion of nuclei. And then the third step is zygote formation. Zygote is a diploid structure. Zygote will be formed, and this zygote will not persist in the life cycle of fungi, because I have just told you that the main body of fungi is haploid, not diploid. That means the fungi do not want this zygote because its body is haploid. So what will happen? As soon as the zygote is formed, meiosis occurs in this zygote. Meiosis or reductional division. Meiosis or reductional division occurs in the zygote, and what are formed? See what I'm writing here. Sexual spores are formed, which germinates to form mycelium of fungi. So I hope you understand now. What were asexual spores? Asexual spores were forming on the mycelium of fungi by mitosis. But how sexual spores are formed? First, during sexual reproduction, there will be fusion of cytoplasm, then there will be fusion of nuclei, then zygote will be formed, but this zygote is not needed by fungi, so meiosis will occur in the zygote and sexual spores will be formed, and they will give rise to a new fungi mycelium. Clear? Now, sometimes just after plasmogamy, karyogamy does not occur, but an intermediate resting stage is seen, called as dikaryophase. Called as dikaryophase. This is important. I have just told you that during sexual reproduction, after plasmogamy, imagine two structures. When they are coming together, their cytoplasm is fused, right? When they are coming together, their cytoplasm, imagine two structures, right? They have genetically different nuclei. These are two structures. When they are coming together, their cytoplasm get fused, but their nuclei are still unfused, right? So this structure is called as the dikaryon. This structure is called as the dikaryon, having two nuclei which are genetically dissimilar, right? Just after plasmogamy, what happens? Karyogamy. That means these two nuclei, they get fused. These two nuclei, they get fused, and they form a diploid nucleus inside it, that is zygote. But sometimes what happens? Sometimes just after plasmogamy, karyogamy do not occur. That means a small resting phase is seen in which there are two nuclei in the same cell, and that is the dikaryophase. So in some fungi, plasmogamy does not occur immediately, karyogamy does not occur immediately after plasmogamy and forms dikaryon, and this dikaryon or dikaryophase is seen only in the members of Ascomycetes This is seen only in the members of Ascomycetes and Basidiomycetes. Right? Then you'll say, ma'am, why not in Phycomycetes? In Phycomycetes, just after plasmogamy, karyogamy occurs. Then you'll ask, ma'am, why not in Deuteromycetes? Because in Deuteromycetes, sexual reproduction is completely absent. Clear? So this intermediate stage between plasmogamy and karyogamy is seen only in Ascomycetes and Basidiomycetes. I hope this is clear to you. See? I hope this is clear. Let's continue with the classification of fungi now. Classification of fungi. We have divided fungi into 4 classes, that is Phycomycetes, Basidiomycetes, Ascomycetes and Deuteromycetes. On what basis we have classified the fungi? First, morphology of the mycelium. It is septate or aseptate. Then fruiting bodies. There are certain structures found in the life cycle called as fruiting bodies in which spores are formed, whether they are present or not. Mode of spore formation, how spore is getting formed in them. These are the line of NCERT, so important. So what is the basis of classification? Morphology of mycelium, then fruiting body, and then mode of spore formation. Now, as I have made a comparative table in algae, which must have made the things easier for you to remember, we will make a comparative table for fungi also. And if you will learn that table, then you will be able to solve many of the questions. So let's make a comparative table. Okay, so let's compare the characters between all the classes of fungi. Let's first compare their common name. Phycomycetes is also called as algal fungi because some of the members of Phycomycetes are found in they are found in the, yes, one more thing I want to tell you here, that further the Phycomycetes is divided into 2 classes. Sorry, that is Oomycetes and sorry beta, yeah, that is Oomycetes and Phycomycetes. Oomycetes and Zygomycetes. I'm a bit tired now. Yeah beta. So the aquatic members of the Phycomycetes, they come under Oomycetes, and the terrestrial members of the Phycomycetes, they come under Zygomycetes. Okay, so the common name of the members which are found in the aquatic habitat, they are the algal fungi because they are present in water like algae. And the Zygomycetes, they are called as conjugation fungi because the sexual reproduction found in them is conjugation. Basidiomycetes are called as club fungi. Why? Because their fruiting bodies are club shaped. The Ascomycetes are called as sac fungi because their fruiting bodies are sac shaped. Deuteromycetes are called as imperfect fungi because the sexual reproduction is absent in them. Right? So these are the general common names. So Phycomycetes have aquatic members also and terrestrial members also, but the rest of the classes, they have only terrestrial members. Okay, now let's discuss their habitat. The members of Phycomycetes are aquatic also, like, right, few members are aquatic also, and terrestrial also, right? The aquatic members, the members of Oomycetes, are generally obligate parasites. Obligate parasites means they can be parasites and nothing else, right? The members which are terrestrial, they are rarely parasites, right? So the members of the Phycomycetes can be aquatic or can be terrestrial. The members of Basidiomycetes, they all are terrestrial. They all are terrestrial. And where are they found? They are found on tree stumps and wooden logs. They are found on tree stumps and wooden logs. Ascomycetes, the members of Ascomycetes are also terrestrial, all of them, and they are found, they can be coprophilous, they can be, parasites, they can be symbiotic, they can be present in symbiotic association, etc., etc. Then this Deuteromycetes is also terrestrial in nature. Deuteromycetes is also terrestrial in nature, but what is important written in NCERT is that they are very important decomposers. You know what are decomposers, right? Which decomposes the organic material, and they help in mineral cycling. They help in mineral cycling. Okay, so this is important. They can be aquatic, they can be terrestrial. Basidiomycetes all terrestrial, generally found on tree stumps. You can see mushrooms on tree stumps, right? Then Ascomycetes, terrestrial. Deuteromycetes, terrestrial. What you have to remember that Deuteromycetes are very good decomposers, and that is why they help in mineral cycling. This question can come directly. Which members of fungi help in mineral cycling? Mostly Deuteromycetes. So let's take the table forward. Now let's compare their mycelium. Compare their mycelium. Obviously, the member of Phycomycetes, Basidio, Asco, and Deutero. So let's compare their mycelium. You know, beta, that the mycelium of Phycomycetes is aseptate, and the mycelium of the members of Basidiomycetes, septate. Ascomycetes, septate. Deuteromycetes, again septate mycelium. Now let's compare the asexual spores found in them. In the class Ascomycetes, the asexual spores were, tell, if you remember, tell. In the members Phycomycetes, zoospores and sporangiospores are seen. Sporangiospores are seen in the terrestrial members, whereas zoospores are seen in the aquatic members. You know that now. Basidiomycetes, sexual spores generally absent. Ascomycetes, sexual spores are ascospores formed by meiosis in zygote. Remember? Oh, sorry, what I've written. Oh, sorry, my God. Oh my God, this is not mistake. This is blunder. Sorry, beta. This was sexual spore. Sorry. So, ascospores, yes, conidia. The asexual spore is conidia. So, what you have learned by the mistake of madam, that is, you do not have to confuse between sexual and asexual spore. Okay? Make this very clear in mind. Zoospore, sporangiospore, and conidia, these are asexual spores. Clear? So, in Basidio, generally absent. In both the classes, conidia, in them, zoospore and sporangiospore. Now, let's compare their sexual spores. The sexual spore of Zygomycetes is zygospore. Sorry. Remember one thing, beta. What is a sexual spore? A sexual spore is either a product of sexual reproduction or formed by meiosis in the zygote. Both are sexual spores. That means zygote is also a sexual spore. Why zygote is a sexual spore? Because it is a product of sexual reproduction. And the spores which will formed by meiosis in zygote, they're also called as sexual spores. Are you clear with the thing I'm saying? Zygote is a sexual spore because it is a product of sexual reproduction. So the sexual spore of Phycomycetes is zygospore. It is the resting zygote. See, it is diploid. Please do not confuse zygospore with zoospore. Please, the question can come. Zygospore is a sexual spore, is an asexual spore. So our eyes will read zygospore, but the mind will remember zoospore. Zoospore is different. Zoospore is the asexual spore, but which is haploid. All the asexual spores are haploid. But here, zygospore is the sexual spore, which is diploid, which is a resting zygote. And ultimately, meiosis will occur in it to form the spores, which will form a new mycelium. In the Basidiomycetes, beta, the sexual spore is basidiospore. And this is haploid. Why this is haploid? Because it is formed after the meiosis in zygote. The sexual spore of Ascomycetes is ascospore, again haploid. And the sexual spore is absent in Deuteromycetes because they do not have sexual reproduction. Clear? Now, let's see what is the condition of sex organs, where the sex organs are present. In Phycomycetes, sex organs are present. In Basidiomycetes, sex organs are absent. In Ascomycetes, present. In Deuteromycetes, absent. Sex organs, you know what are sex organs, right? Sex organs are present only in Phycomycetes and Ascomycetes. That means they are absent in Basidiomycetes and Deutero. In Deutero, it is understood because sexual reproduction is absent. But why in Basidiomycetes? Remember one thing, beta. I'll tell you this later on after the table is completed, that Basidiomycetes do not have sex organs, but still they can reproduce sexually with the help of somatogamy. I'll just tell you this. Okay. Now let's move further with the table. Now let's compare sexual reproduction, where the sexual reproduction is present. Sexual reproduction is present in Phycomycetes. It is present in Basidiomycetes. It is present in Ascomycetes, but it is absent in the Deuteromycetes. Then let's compare the condition of fruiting bodies. Which fungi has fruiting body? Fruiting bodies are absent in Phycomycetes. They are present in Basidiomycetes and they are called as basidiocarp. Fruiting bodies are present in Ascomycetes and called as ascocarp, and it is absent in case of Deuteromycetes. Remember one thing, beta. Wherever there is dikaryophase, that means wherever there is formation of dikaryotic mycelium, only there fruiting bodies can be formed, because fruiting bodies are formed on dikaryotic mycelium. So remember, all those fungi which have dikaryotic mycelium, they have fruiting body, and you know that dikaryotic mycelium is found only in Basidio and Asco. That is why fruiting bodies are found only in Basidio and Asco, absent in Deutero and the Phycomycetes. Okay, you'll remember this. This is important. Okay, now let's discuss some examples over here. So let's first remember the table, the whole of the table up till here. It is pretty important. Yeah. So what we have discussed up till now, beta, we have discussed that the members of Phycomycetes can be aquatic or can be terrestrial. The aquatic members are Oomycetes and the terrestrial members are Zygomycetes. Okay. And the aquatic members of Phycomycetes are generally parasites, and the terrestrial members, that is Rhizopus and Mucor, they are rarely parasites, they are saprophytes. Rest all the fungi, they are terrestrial, and they are the Deuteromycetes best decomposers, help in mineral cycling. Okay, now there, learn this table along with me. Except Phycomycetes, rest all the three has septate mycelium. Asexual spore. In Phycomycetes, zoospore, sporangiospore. Asexual spore generally absent in Basidiomycetes, Ascodeuteroconidia. Sexual spore, zygospore. It is not zoospore, and it is 2n, not n, because it is resting zygote. Then meiosis will occur after some time, and then spores will be formed. Sexual spore of Basidiomycetes is Basidiospore. Ascospore, absent, right? Sex organs are absent in Basidiomycetes and in Deuteromycetes. But even after the absence of sex organs, sexual reproduction is present in Basidiomycetes. Sexual reproduction is absent only in case of Deuteromycetes. Fruiting body are present only in Basidio and Ascomycetes. That is Basidiocarp and Ascocarp. If you will learn this table, you can solve many questions. Okay. Now let's discuss those points which are not there in the table. Let's first discuss the important points of Phycomycetes. Okay. The examples of Phycomycetes given in NCERT is albugo. Albugo is an obligate parasite. Albugo is an obligate parasite. That means it can only be parasite and nothing else. It is a parasitic fungi which cause white rust, in mustard. This is very important and can come in NEET, which cause white rust in mustard. Generally, it is thought that rust-causing fungi are Basidiomycetes. It is true, but remember, that is black rust. This is white rust. White rust in mustard is caused by Phycomycetes albugo. Then, another members are Rhizopus. This albugo is the aquatic member. Rhizopus, Mucor, these are the terrestrial members, and both of them are saprophytes and rarely parasites. Okay, and what NCERT is writing about them? In their life cycle, a resting zygote, that is zygospore, is formed. And you know what is zygospore? It is resting zygote. Clear? Now let's discuss some extra points regarding the Basidiomycetes. I have just told you, beta, that Basidiomycetes has dikaryotic mycelium in its life cycle. Remember, it has dikaryotic mycelium in its life cycle. How dikaryotic mycelium gets formed in its life cycle? This dikaryotic mycelium, which is formed after plasmogamy, right? After plasmogamy, it develops some structures on it. And these structures are called as ascus. These are called as ascus. Just go with me line by line and you will understand. the dikaryotic mycelium in the life cycle of Basidiomycetes develops some structures. Oh, what is happening? Develops some structure, club-shaped structures. Sorry, beta, I'll correct it. I am a bit tired, I think. Yeah. So they form some club-shaped structures, and these club-shaped structures are called as basidium. Basidium, or the plural form is called as basidia. Okay, ta? So these structure come out of dikaryotic mycelium, right? And the dikaryon, the two nuclei, they fall in this structure, and ultimately the karyogamy occurs inside basidium. Karyogamy, that is fusion of nuclei. Karyogamy occurs inside basidium. So this can be a direct question, that what is the site of karyogamy in Basidiomycetes? Your answer will be basidium. So when karyogamy will occur inside these basidium, what will be formed, beta? What will be formed? Zygote. After karyogamy, zygote will be formed. Where? Inside the basidium. And you know that this zygote will undergo meiosis. This zygote will undergo meiosis. Right. And what will be formed after meiosis? The sexual spores. But they are formed on the surface. So on the surface of the basidium, the sexual spores, that is basidiospores, are formed after meiosis in zygote. So what is the site of meiosis? Meiosis occurs inside basidium. This direct question comes. Where karyogamy occurs in Basidiomycetes? Answer: basidium. Where meiosis occurs in Basidiomycetes? Answer: basidium. So remember, site of karyogamy and site of meiosis. Site of karyogamy and site of meiosis, both are basidium in Basidiomycetes. Clear, beta? You can imagine dikaryotic mycelium in the Basidiomycetes. Some club-shaped structure arises called as basidium. Inside basidium, ultimately fusion of nuclei happens. Then zygote gets formed. Then in that zygote, meiosis occurs, and ultimately the basidiospores are found on, not inside, on the surface of the basidium, right? So basidiospores are the sexual spores. Clear? Basidiospores are the sexual spores. So what you will say regarding basidiospores? First of all, they are haploid, obviously found by meiosis in zygote. Then they are exogenous. Remember, exogenous, because they are formed on the basidium. Okay, remember this point. This is very important. And this is, you know, the sexual spore of Basidiomycetes. Now, what is fruiting body? I was telling you what is fruiting body. The fruiting body of Basidiomycetes is called as basidiocarp. The fruiting body is basidiocarp. And how it is formed? It is formed by the aggregation of many dikaryotic hypha. When many dikaryotic hypha which have basidium on them, they aggregate together. So aggregation of many basidia, so when many basidia, they aggregate together, they form the fruiting body, that is basidiocarp. Okay, so I hope you can imagine now. When you close your eyes, you will see the dikaryotic mycelium. From that dikaryotic mycelium, basidia are arising out. They are falling from the dikaryotic mycelium. Inside them, zygote will form, right? Karyogamy will occur. Then meiosis will be there. Then basidiospores are formed on the surface of basidia, which are sexual spores, right? And the aggregation of all these basidia is the fruiting body, that is the basidiocarp. Now I have just told you that in Basidiomycetes, sex organs are absent, right? What is absent in Basidiomycetes? Asexual spores are absent, asexual spores, and sex organs are absent, right? So how asexual reproduction occurs in Basidiomycetes? By fragmentation. This is given in NCERT, right? Asexual spores are absent, but how asexual reproduction occurs in them? By fragmentation. Clear? Now in Basidiomycetes, sex organs are absent, but still the members of Basidiomycetes, they perform sexual reproduction. They perform sexual reproduction. Sex organs are absent, but sexual reproduction is present. Why? Because that is done with the help of, because sexual reproduction is done by somatogamy. Is done by somatogamy. And what is somatogamy? In somatogamy, two somatic hypha. What is somatic hypha, beta? Somatic means which is not reproductive in nature. That is somatics. So two somatic hypha, they come together. This is also a somatic cell. This is also a somatic. Somatic cell is a cell which is not germinal in nature. So they will fuse. The nucleus of this is suppose this, and they are genetically different. This somatic hypha belongs to plus strain. This somatic hypha belongs to minus strain. They are genetically different. These two somatic hypha, they fuse with each other. They fuse with each other. Okay, they fuse with each other to form this. This is dikaryon. To form this is dikaryon, and this whole hypha formed by somatogamy will be the dikaryotic mycelium, will form the dikaryotic mycelium. And on then this dikaryotic mycelium, then ultimately what will happen? Basidia will come out. So these are monokaryotic somatic hypha. These are monokaryotic somatic hypha. You know what is monokaryotic, right? So these are monokaryotic hypha. They are fusing with each other and forming the dikaryotic mycelium, on which ultimately fruiting bodies will be formed. So here, when there is fusion of two somatic cells to form a dikaryon, and here only plasmogamy is occurring. Only plasmogamy occurs. Karyogamy occurs inside basidium later on. Remember, when the basidia arise from dikaryotic mycelium, karyogamy occurs in the basidium. So can you imagine? In Basidiomycetes, sex organs are absent, but they can sexually reproduce with the help of a process called as somatogamy. Somatogamy, in which sex organs are not required, but two somatic hypha, two monokaryotic, you know what is monokaryotic now. So two monokaryotic somatic hypha, they come close to each other, they fuse and form a dikaryotic mycelium. And during somatogamy, only plasmogamy is seen. Only fusion of cytoplasm. You can see there is no fusion of nuclei. Fusion of nuclei will occur inside the basidium to form zygote. Clear, beta? So this is important because this is given in NCERT. What is somatogamy? During somatogamy, only plasmogamy is seen. And this is the way how Basidiomycetes reproduce sexually without having sex organs. Okay? Now, some examples of the members of Basidiomycetes. Some examples are Puccinia, which is called as the rust fungi because it causes the disease rust in cereals, but that rust is either black or brown rust. White rust was caused by albugo. Okay. Yeah. Now, second member is Astilago. Astilago is called as the smut fungi because it causes the disease smut. So both of them are parasites. Then another members are puffballs. Puffballs, also called as Lycoperdon. Another members are mushroom. And the scientific name of mushroom is Agaricus. And this is the edible fungi. You eat mushroom, right? So you eat mushroom is Agaricus. Mushrooms. Then bracket fungi, also called as Ganoderma. So these all are given in NCERT. Okay. So these are some famous members: Puccinia, Astilago, Lycoperdon, mushroom, bracket fungi. Clear? Now let's discuss some important points of Ascomycetes. Yes. Ascomycetes, again, what happens? That dikaryotic mycelium is formed in their life cycle, like the Basidiomycetes. You know that dikaryotic mycelium is found in the life cycle of Ascomycetes and Basidiomycetes. So out of this dikaryotic mycelium, what arises is sac-shaped structures, and these are called as ascus. Same as that of the Basidiomycetes, but there we used to call them the club-shaped Basidia, but here they are called as sac-shaped ascus, and the plural term is called as SI, right? Then you know what will happen. Karyogamy, right? Karyogamy will occur inside the ascus. So after karyogamy, what will formed inside the ascus? Obviously zygote. Then after the formation of zygote, what will happen, beta? Meiosis. So inside the ascus, meiosis will occur, and what will be formed? Ascospores. But these ascospores are formed inside the ascus. I hope you can make out the difference. There in the Basidiomycetes, the basidiospores were formed on the surface of the basidium. That is why there were exogenous sexual spores. But ascospores are endogenous sexual spores, right? So they are endogenous. Why? Because they are formed inside the ascus and they are sexual spores of Ascomycetes. Okay, beta. So now what we can write, that site of karyogamy and meiosis. Site of karyogamy and meiosis in Ascomycetes is ascus. In Basidiomycetes, the site of karyogamy and meiosis was basidium, but here it is ascus. I hope that the difference is clear. Ascospores are the sexual spores found inside ascus. Basidiospores were forming outside the basidium. Clear? And the aggregation of ascus, aggregation of SI, the aggregation of many SI is called as the fruiting body. The fruiting body, that is ascocarp. That is ascocarp, right? The fruiting body of Basidiomycetes was basidiocarp. Okay, now let's take some examples, important examples of ascomycetes, and they are yeast. You know that yeast is a unicellular member of ascomycetes, and you know what is the function of yeast. It is used in bakery industry and the alcohol industry. Second example is penicillium. You know what is the importance of penicillium. The first antibiotic, penicillin, was formed from it. Then another important example is aspergillus. Aspergillus. Then another important example is neurospora. Neurospora is called as the Drosophila of plant kingdom because many genetic research has been done on neurospora. Okay. Then comes another important member is truffles and morels. This question has come in NEET. Truffles and morels, right? Both of them are edible members of ascomycetes. This has come in NEET. This was the question of NEET given in NCERT. Truffles and morels are the members of ascomycetes which are edible and eaten. Okay, so these are some important members of the ascomycetes class. Now let's move on to the deuteromycetes. What is special regarding deuteromycetes? You know, they are important in maintaining the mineral cycle, sexual reproduction, sex organs absent. Clear? Now, the members of deuteromycetes, beta, some of the members of deuteromycetes, all the members which did not show any sexual reproduction, they were placed in deuteromycetes. But when it was seen that some of the forms, they show sexual reproduction, then they were migrated either to Basidiomycetes or to ascomycetes. So some of the members which later on, this is the line of NCERT, some of the members which later on showed sexual reproduction were migrated to either ascomycetes or Basidiomycetes. Showed sexual reproduction were later on migrated to ascomycetes or Basidiomycetes. Okay. Now some examples of the members of deuteromycetes. Important examples are Alternaria. Alternaria is the member which cause early blight of potato. Is cause the disease early blight of potato. Another important member is Coletotrichum. Coletotrichum causes red rot of sugarcane. Red rot of sugarcane. Helminthosporium. This fungi was responsible for the Great Bengal famine, came in 1943 during the freedom time. This destroyed the rice, so it causes leaf spot of rice and was responsible for the Great Bengal famine. It causes leaf spot of rice. Okay. And trichoderma. Trichoderma causes skin diseases. So these are some important members. Learn with me. Alternaria, Coletotrichum, Helminthosporium and Trichoderma. Clear? So this is the end of deuteromycetes. So we have finished fungi. I hope things are clear to you. Beta, what are asexual spores? What are sexual spore? Right? What is zygospore? Right? Comparative table you have to learn in the fungi. Then important points. Sex organs and asexual spores both are absent in Basidiomycetes. In Deuteromycetes, what is absent? Sexual spores and sexual reproduction both. Okay, so learn the table and your fungi will be ready. Hope this is clear to you. So let's move on further. Now comes the virus. As you know, beta, that virus is considered as, let me check my mic. Yeah, full battery. So, yeah, where I was, yeah, viruses, they are considered as non-cellular or acellular organisms. That means they do not have a proper cell, right? And that is why they did not get any place in the 5-kingdom system of classification, right? So remember this. And some of the characters they have are living characters, like presence of a protein coat, like presence of nucleic acid. They can cause infections. These are some living characters of viruses. And they are, you know, they do not have a proper cell. They do not have respiratory enzymes. They cannot grow outside the host cell. These are some non-living characters. So that is why they are taken as connecting link between living and non-living organisms. Okay. Now, there are some important scientists related with virus, and they are important. The first one is the Louis Pasteur. Louis Pasteur gave the term viruses. Okay, I think there is some misprinting in the new NCRT. Please kindly correct it. He gave the term virus. The first scientist to discover the virus, he just gave the term virus. The first scientist to discover the virus is D.J. Ivanovsky. He first discovered the virus, and whose name was tobacco mosaic virus, that is TMV. And he also discovered the filterable property of viruses. He told that viruses are smaller than bacteria and they cross the bacteriological filter because they are smaller than bacteria. Okay, so what two important things DJ Ivanovsky done? The name is interesting, DJ Ivanovsky. So he first discovered the tobacco mosaic virus and he defined the filterable property of viruses. Then came Beijerinck. Beijerinck said that they are living, like viruses are living in nature, which are contagious, infectious, and they are fluid-like structures. So he called them contagium vivum fluidum, that is living infectious fluids. And then came W.M. Stanley. He first crystallized the viruses. Viruses can be crystallized, they can be converted into the form of a crystal, and he crystallized the first virus, that is tobacco mosaic virus. So these are the important scientists from the point of view of NEET. Okay. Okay, then let's move on to the structure of virus now. If I talk about the structure of virus, beta, if I talk about the structure of virus, it has just simply two things: the protein coat, which is also called as capsid, right? The protein coat is called as capsid, which is composed of many smaller subunits called as capsomeres. So the protein coat is composed of many smaller subunits called as capsomeres, and this protein coat constitutes about 95% part of the structure of virus, and rest it has the nucleic acid, the genetic material, right? And it just constitute 5% part of the structure of virus, and the genetic material of viruses can be RNA or can be DNA. Never, a virus will never have, see, this is a question which has come in NEET, a virus has never both DNA and RNA. Never, either DNA or RNA. So it has a protein coat around it, which is 95% of the structure, called as capsid, composed of capsomeres. It has nucleic acid, or the genetic material, which can be either DNA or RNA. In a single virus, both of them can never be present together, either DNA or RNA. Okay. So on the basis of this, you can say that some of the viruses are RNA viruses and some of the viruses are DNA viruses. Now let's classify the viruses based on the host. Classification of virus based on the host. Right. So based on the host, viruses can be phytophagony or plant viruses. Plant viruses which cause diseases in plants. Right. Then animal viruses which causes diseases in animals. And then comes the bacteriophages, which causes diseases in bacteria. Okay. So see beta, plant viruses are single-stranded RNA viruses. Remember this. If someone asks that what is the nature of tobacco mosaic virus, what you will say? Tobacco, it's a plant, so obviously it will be having single-stranded RNA. So remember, all the plant viruses are single-stranded RNA viruses, right? Animal viruses can be single-stranded RNA viruses. They can be double-stranded DNA viruses, or they can be double-stranded RNA viruses. That means which category is not here? That is single-stranded DNA. Single-stranded DNA is not here. This is given in NCERT, important. Animal viruses can be single-stranded RNA, double-stranded DNA, or double-stranded RNA. Okay, then bacteriophages are mostly double-stranded DNA. Not always, but mostly the bacteriophages are double-stranded DNA viruses. Clear? This is important. Bacteriophages are bacterial viruses. Clear? So this is very important beta. This is very important because it is given in NCERT. So let's take some example of single-stranded RNA viruses. In case of animals, you know that all the plant viruses are single-stranded. Tobacco mosaic virus, papaya curl virus, cucumber mosaic virus, these all will be the single-stranded RNA viruses, right? But if I take example of some animal viruses which are single-stranded, then it can be measles, as it is given in NCERT, important. Measles, mumps, right? Rabies, polio, HIV, that is AIDS, SARS, severe acute respiratory syndrome, corona, right? The COVID, right? These all are single-stranded RNA viruses. Now, if you want to take some double-stranded DNA viruses, in the double-stranded DNA viruses, herpes, you know the disease herpes, smallpox. These diseases are given in NCERT, so it becomes important. Cowpox, not cowpox, sorry, chickenpox. Chickenpox. Okay, so these are double-stranded DNA viruses. Okay, beta. So remember these things. All the plant viruses, single-stranded RNA viruses. Then these are some single-stranded animal viruses. These are double-stranded animal viruses. Okay. So these are given in NCERT, learn them. Corona, which has, you know, corona is a single-stranded RNA virus which has caused so much of havoc and highly mutable. Viruses are very highly mutable because of the proteinaceous structure they have. They are highly mutable, and the RNA they have. Okay, now let's discuss Subviral agents. What is subviral agent? A subviral agent is an agent in which the virus is not properly complete. I have just told you that a virus has a genetic material and protein code. But something would be absent from subviral agent. That means they are not complete viruses. They are not complete viruses. So these kind of viruses are called as subviral agents. And the two types are, one is viroids, and the second are prions. Viroids and prions. Now, in viroids, protein coat is absent. In viroids, protein coat is absent, and in prions, nucleic acid is absent. Genetic material is absent. So something is absent from them, right? The viroids, they are also called as free RNA particles. Viroids are also called as free RNA particles. Why? Because their genetic material is not surrounded by any protein coat, and they are RNA viruses. They are RNA viruses, and their RNA is extremely small. Their RNA is extremely small. On the other hand, the prions are called as proteinaceous infectious particles. They are called as proteinaceous infectious particles. Why? Because they do not have the genetic material and their protein is infectious in nature, which cause the disease. So in viroids, protein coat absent, in them nucleic acid absent. They have extremely small RNA as their genetic material. That is why they are free RNA particles, and their protein coat causes the disease. Now let's discuss more about viroids. Viroids have low molecular weight. Why low molecular weight? Because 95% of the part, that is protein coat, is absent, and their RNA is also extremely small, so they have low molecular weight. Another important point, who discovered the viroids? T.O. Diner. This has come in NEET. T.O. Diner discovered viroids. T.O. Diner discovered viroids, okay? And they cause diseases only in plants. Viroids cause diseases only in plants, not in animals. So they are plant viruses, and they cause 20 different diseases in plants. They cause 20 different diseases in plants, right? One of the important disease caused by viroid is potato spindle tuber disease, PSTD. Potato spindle tuber disease. Okay. Then let's talk about prions. Prions are animal viruses. They cause diseases in animals. And what kind of diseases? They cause a special kind of diseases called as neurodegenerative diseases. They cause neurodegenerative diseases in animals. What are neurodegenerative diseases? The prions, they attack our brain cells, nerve cells, and these kind of diseases called as neurodegenerative diseases. So these diseases can occur in cattle or can occur in humans. In cattle, these disease is called as BSE, that is bovine. It is written in NCERT, so becomes important. This is bovine spongiform encephalopathy, or this is also called as mad cow disease. This is also called as mad cow disease. In humans, it is called as the Creutzfeldt-Jakob disease, right? But both of them are neurodegenerative diseases. Both of them are neurodegenerative diseases. Okay, so let's revise it fast. Viroids. What is the difference between what is the difference between viroids and prions? Viroids, they do not have protein coat. Prions, they do not have nucleic acid. The molecular weight of viroids is very low as compared to the normal viruses. But the molecular weight of prions is similar to the viruses because they have 95% part, that is protein in them. Okay. The viroids can cause disease only in plants. That is why they are plant viruses. Okay. And some of the disease are potato spindle tuber disease. But prions causes diseases animals. That is neurodegenerative disease. Okay, mad cow disease in cattle and Creutzfeldt-Jakob disease in humans. So these are the differences between prions and viroids. Okay, so yes, this is important. Remember these things. Now, these are the structures given in NCERT which are important. This is the diagram of tobacco mosaic virus. I hope you will identify the diagram of tobacco mosaic virus. This is the capsid or the protein coat, and this is the single-stranded RNA. This is the single-stranded RNA which is arranged helically. This is the diagram of T2 bacteriophage. Okay, T series, either T3 or T4, same, right? So T2 bacteriophage has this type of structure. This head, the shape of the head is polyhedral or prismatic. The shape of the head is polyhedral or prismatic shape, right? Prismatic or polyhedral shape of the head. This head contains what kind of genetic material? It contains double-stranded linear DNA. It contains, let's write with red, it would be more visible. Yeah, this head contains double-stranded linear DNA. Okay, so we divide the structure of a bacteriophage, a T2 bacteriophage, into two categories: head and tail. This part is head, this part is tail. The head, the shape of the head is polyhedral and it contains double-stranded linear DNA. This is the tail, and this is the collar which connects tail with the head. The tail, the end point of tail contains a hexagonal plate having 6 ends, and from that hexagonal plate 6 tail fibers come out, and these tail fibers help the virus to attach to the bacterial cell. Okay, so this is all about virus. I hope you understood the whole biological classification well, and what you have to do now, beta, listen the lecture carefully. I have covered all the points of NCERT. No point of NCERT is left. Listen to the lecture carefully, then read your NCERTs line by line carefully, and then do questions, and your chapter would be ready. I hope this would help you in best manner. Thank you.